Hello there. Welcome to Relax with Science, where the intricate poetry of the universe lulls you to sleep. You have had a long day. I know. It takes so much energy just to keep up with the waking world, but that part of the day is over now. You can let it all go. Allow yourself to feel exactly how tired you truly are. Let your shoulders drop away from your ears. Unclench your jaw. Let your tongue fall away from the roof of your mouth and feel the heavy, comforting warmth of your body sinking softly and deeply into the bed below you. You are safe here. There is nowhere else you need to be. Tonight, we are going to embark on a gentle, fascinating journey inward, exploring the quiet, chemical rivers that flow within you. We will be traveling through the endocrine system. As we wander through this microscopic world, please remember, you do not need to memorize a single fact. There is no test to pass, and nothing you need to hold on to. Simply let my voice wash over you and listen to the quiet perfection of the science as it softly hums inside your very own body. Right now, without any instruction from you, without any effort or conscious thought, something beautiful is moving through you. A gentle tide of molecules traveling through the quiet rivers of your arteries, in the tiniest, delicate vessels of your bloodstream, finding their way with a precision that feels almost impossible, but is simply the ordinary, miraculous fact of being alive. These are your hormones. The slow, steady, deeply transformative language of your body. While your nervous system flashes like quick, bright lightning, your endocrine system is the shifting of the seasons. It does not demand immediate action. It is the slow, patient orchestration of growth, of healing, of your body's long, peaceful relationship with time. A hormone released into your bloodstream may take hours to reach its destination, but when it arrives, its gentle influence lingers. Tonight, we are going to travel along these quiet pathways together, through the soft rhythms and the delicate feedback loops that make up your body's inner harmony. And I want to offer you this thought. As you drift deeper into the mattress, everything we talk about tonight is already happening inside you. The cortisol that kindly helped carry you through your busy afternoon is fading now, making way for the melatonin, which is, even as I speak these words, rising in your bloodstream, whispering to every cell in your body that it is dark, that you are safe, and that it is time, at last, to rest. The insulin that quietly balanced your evening, and the growth hormone that will soon peak to heal and restore you in the deep, heavy sleep you are approaching. All of it is yours. We are simply giving it names. I wonder where you are right now, what kind of day your body carried you through to arrive here in this quiet moment. Whether it was a long day or a short one, a day that demanded a great deal of your energy, or one that was kinder. If you feel drawn to it, and only if you wish, let me know in the comments where you are resting tonight. Just a city, a country, a time on the clock, or the soft temperature of the midnight air outside your window. I read every one. It matters to me that you are there, wherever there happens to be. Let the room be quiet now. Let your breath slow, in and out. And let your blood carry its soothing messages, as it has done every night of your life, into the waiting dark. If these words bring you any ease tonight, any settling of the day's remaining weight, a quiet like on the video is a small and generous way to let me know. It helps these stories find their way to someone else who might need exactly this kind of company in the later hours. If you know someone who carries the exhaustion of a long day to bed with them, consider sending this to them. No explanation necessary. Just a pathway left somewhere they will find it. And if there is ever something that could be softer, slower, or more beautiful about what we make here, I genuinely want to hear it. This space belongs to the person lying in the quiet. It has always been for you. Close your eyes. Drift. And let the universe within you do the rest. The endocrine system is the body's primary, long-range chemical communication network, operating through approximately 50 known hormones, secreted by specialized glands and tissues, into the bloodstream, where they travel to target cells throughout the body and produce effects that last from minutes to years, making it, in its temporal range, the most expansive signaling system the body possesses. The word hormone comes from the Greek hormon, meaning to set in motion, and this etymology is a good beginning for understanding what hormones do and why the endocrine system exists. The nervous system, with its electrical signals and its millisecond timescales, is the system of immediate action, the system that moves the hand from the hot surface that adjusts the balance of the body in the fraction of a second before a fall, that processes the sensory world at the speed required to navigate it. But there is a whole other category of biological process that cannot be managed on this timescale. The growth of the skeleton through childhood requires not a single adjustment, but a sustained, years-long program of bone elongation, driven by continuous hormonal signals. The reproductive cycle requires a monthly choreography of hormonal rises and falls, coordinated across the hypothalamus, the pituitary, the ovaries, and the uterus. The metabolic response to fasting, the shift from burning glucose to burning fat, the conservation of protein, the maintenance of blood sugar during the hours without food, requires a sustained hormonal reconfiguration that unfolds over hours and must be maintained for as long as the fast continues. The endocrine system is the body's instrument for all of these sustained, gradual, long-range biological programs. Its signals travel slowly. A hormone released from the adrenal gland reaches the kidney in approximately 30 seconds via the bloodstream, but they persist, maintaining their influence on target tissues for as long as they remain in the circulation and their receptors remain responsive. The 50 or so hormones identified so far are themselves the product of decades of discovery, and the number continues to grow as researchers identify previously unknown secreted molecules from the heart, the fat tissue, the gut, the muscle, with hormonal functions. The endocrine system is larger and more distributed than the classical picture of a few specialized glands suggests. The hypothalamus, a small region at the base of the brain, weighing approximately 4 grams, serves as the master conductor of the endocrine system, translating neural signals into hormonal commands and governing the activity of the pituitary gland through a cascade of releasing and inhibiting hormones secreted into the portal blood vessels of the pituitary stalk. The hypothalamus is, in the architecture of the endocrine system, the place where the nervous system and the hormonal system become one. It is a brain region containing neurons organized into nuclei, receiving and sending electrical signals like any other part of the central nervous system. But many of its neurons are also secretory cells, neuroendocrine cells, whose axon terminals release peptide hormones, not into a synapse but into the bloodstream, using the vascular system as their signal pathway. This dual nature makes the hypothalamus the interface between the two great communication systems of the body, the instant electrical language of neurons and the slow chemical language of hormones. The hypothalamus governs the pituitary gland, the grape-sized structure hanging below it, at the end of the pituitary stalk, by secreting a set of releasing and inhibiting hormones into a specialized portal circulation that flows directly from the hypothalamus to the anterior pituitary. These signals tell the pituitary which of its own hormones to release and in what amounts. Thyrotrophin-releasing hormone tells the pituitary to release thyroid-stimulating hormone. Growth hormone-releasing hormone tells the pituitary to release growth hormone. Corticotropin-releasing hormone tells the pituitary to release adrenocorticotropic hormone, which will tell the adrenal glands to produce cortisol. The hypothalamus is also directly connected to the posterior pituitary, not through portal blood, but through the direct axons of hypothalamic neurons that terminate in the posterior pituitary and release their hormones, oxytocin and antidiuretic hormone, directly into the systemic circulation. The 4-gram hypothalamus receiving inputs from virtually every region of the brain, from the limbic system, the cortex, the brainstem, the retina, and integrating them into the hormonal commands that govern metabolism, reproduction, growth, stress response, and water balance, is perhaps the most consequential small structure in the body. The pituitary gland, sometimes called the master gland, is divided into two functionally distinct lobes, the anterior pituitary and the posterior pituitary, which differ in their embryological origin, their mode of hormonal release, and the hormones they produce, and which together secrete 9 of the most important hormones in the body. The designation of the pituitary as the master gland reflects its position in the hormonal hierarchy. It receives commands from the hypothalamus above and issues commands to other endocrine glands below, sitting at the center of the body's major hormonal axis. But the two lobes of the pituitary are so different in their origins and mechanisms that they are, in some ways, two distinct organs sharing an anatomical home. The anterior pituitary, the adenal hypophysis, develops from an upward evagination of the roof of the embryonic mouth, a pocket of epithelial tissue called Rathke's pouch that migrates upward during development to meet the downward growth of the diencephalon. It is a glandular tissue containing 5 distinct cell types, each producing a specific hormone under the control of hypothalamic releasing and inhibiting signals, somatotrophs producing growth hormone, corticotrophs producing adrenocorticotropic hormone, thyrotrophs producing thyroid-stimulating hormone, gonadotrophs producing follicle-stimulating hormone and luteinizing hormone, and lactotrophs producing prolactin. The posterior pituitary, the neurohypophysis, is a fundamentally different structure. It is not a gland at all, but a direct extension of the hypothalamus, a bundle of axon terminals belonging to neurons whose cell bodies sit in the hypothalamic supraoptic and paraventricular nuclei. The hormones it releases, oxytocin and antidiuretic hormone, are not made in the posterior pituitary, but are synthesized in the hypothalamus, packaged into vesicles, and transported down the axons to the posterior pituitary, where they are stored and released on demand. The pituitary gland, sitting in its bony cradle, the sella turcica of the sphenoid bone, is the hormonal command center of the body. But it is, ultimately, a relay station for the hypothalamus. The hypothalamus commands. The pituitary executes. The thyroid gland, the butterfly-shaped structure in the anterior neck, weighing approximately 25 grams, produces the thyroid hormones, thyroxine and triiodothyronine, which regulate the metabolic rate of virtually every cell in the body, and whose deficiency or excess produces some of the most wide-ranging clinical syndromes in medicine. The thyroid hormones, T4, thyroxine, containing 4 iodine atoms, and T3, triiodothyronine, containing 3, are unusual among hormones in their requirement for a dietary mineral, iodine, for their synthesis. The thyroid follicular cells absorb iodide from the blood with extraordinary efficiency, concentrating it to levels 25 times higher than the plasma, and use it to iodinate the tyrosine residues of thyroglobulin, a large protein that forms the scaffold for thyroid hormone synthesis and storage in the colloid-filled follicles of the gland. The coupling of two iodinated tyrosine residues produces either T3 or T4, which are released into the bloodstream when the thyroid is stimulated by TSH. In the blood, thyroid hormones travel predominantly bound to carrier proteins, primarily thyroid-binding globulin, with only a tiny fraction as free, biologically active hormone. In target cells, T4 is converted to T3 by deiodinase enzymes, T3 being the more potent, shorter-acting form that actually binds to the nuclear thyroid hormone receptors and regulates gene transcription. The range of processes regulated by thyroid hormones is almost impossibly broad. They control the basal metabolic rate, the rate at which every cell consumes oxygen and produces energy at rest, and they regulate body temperature, heart rate, gastrointestinal motility, mood, cognition, and the development of the brain in the fetus and neonate. Hypothyroidism, the underproduction of thyroid hormones, produces the characteristic picture of a slowed metabolism, fatigue, cold intolerance, weight gain, constipation, dry skin, slow heart rate, cognitive slowing, and depression. Hyperthyroidism produces the opposite, heat intolerance, weight loss despite increased appetite, palpitations, anxiety, tremor, and diarrhea. The thyroid is, in the most literal sense, the body's metabolic thermostat, set by the hypothalamus and the pituitary, turning the rate of cellular life up and down in response to the body's long-term metabolic needs. Insulin, the peptide hormone produced by the beta cells of the islets of Langerhans in the pancreas, is the body's primary signal for glucose uptake and storage, and its discovery in 1921 by Frederick Banting and Charles Best was one of the most consequential moments in the history of medicine, converting type 1 diabetes from a failed diagnosis to a manageable condition within months. Before insulin was discovered, a diagnosis of type 1 diabetes mellitus, the form caused by autoimmune destruction of the pancreatic beta cells, was a death sentence, typically measured in months for children and young adults. The metabolic catastrophe of insulin deficiency, the failure of glucose to enter cells, the resulting cellular starvation in the face of abundant blood glucose, the compensatory breakdown of fat and protein, the production of ketone bodies in quantities that acidified the blood to fatal levels, was relentless and irreversible. In 1921, Frederick Banting, a Canadian surgeon with no prior research experience, and Charles Best, a medical student working in John MacLeod's laboratory at the University of Toronto with the biochemist James Collip, succeeded in isolating a pancreatic extract that could lower blood glucose in diabetic dogs, and, by January 1922, in the first human patient, a 14-year-old named Leonard Thompson, who was dying of diabetes, and who, within days of receiving the insulin injections, began to recover. The transformation was witnessed and described in terms that approached the miraculous young people at death's door, who, within days of receiving insulin, were sitting up, eating, speaking, returned to life by a molecule that their bodies had ceased to produce. Banting and MacLeod received the Nobel Prize in Physiology or Medicine in 1923. Insulin itself is a relatively small protein, 51 amino acids, arranged in 2 chains linked by disulfide bonds, and it acts primarily by binding to the insulin receptor on the surface of muscle cells, fat cells, and liver cells, triggering a signaling cascade that drives the glucose transporter GLUT4 to the cell surface, allowing glucose to enter. It also promotes the storage of glucose as glycogen in the liver and muscle, and the synthesis of fat from excess glucose in the adipose tissue. Glucagon, the hormone produced by the alpha cells of the pancreatic islets, structurally related to insulin but functionally its opposite, raises blood glucose by stimulating the liver to break down glycogen and to synthesize new glucose from non-carbohydrate precursors. And the ratio of insulin to glucagon in the portal circulation is the primary signal governing the liver's metabolic state. Insulin and glucagon are the yin and yang of blood glucose regulation, opposing hormones from adjacent cells in the same organ, each released in response to the nutritional state that the other suppresses, together maintaining blood glucose within the narrow range of approximately 4 to 6 millimoles per liter that the brain requires for normal function. After a meal, rising blood glucose stimulates the beta cells to release insulin and suppresses the alpha cells' release of glucagon. The high insulin to glucagon ratio signals the liver to absorb glucose from the portal blood, to synthesize glycogen, and to suppress glucose production. Between meals, falling blood glucose suppresses insulin release and stimulates glucagon release. The high glucagon to insulin ratio signals the liver to break down glycogen, glycogenolysis, releasing glucose into the blood, and, if the fast is prolonged, to synthesize new glucose, gluconeogenesis, from amino acids, glycerol from fat breakdown, and lactate recycled from peripheral tissues. Glucagon is a 29 amino acid peptide that binds to a G protein-coupled receptor on hepatocytes, triggering an increase in cyclic AMP that activates protein kinase A, phosphorylating and activating the enzymes of glycogenolysis and gluconeogenesis, while simultaneously inhibiting the enzymes of glycogen synthesis and glycolysis. The liver, the body's metabolic hub, is governed in its second-to-second chemistry by the insulin to glucagon ratio, which it reads from the portal blood that drains the intestine and the pancreas and arrives at the liver before reaching the rest of the circulation. The liver has the first reading of the hormonal message. It responds before any other organ knows the nutritional state has changed. Cortisol, the primary glucocorticoid secreted by the zona fasciculata of the adrenal cortex in response to adrenocorticotropic hormone from the pituitary, is perhaps the most versatile hormone in the body, affecting the metabolism of carbohydrates, proteins, and fats, modulating immune function, maintaining vascular tone, and providing the essential permissive background for many other hormonal responses. Cortisol is the hormone of sustained challenge, not the hormone of the immediate stress response. That is adrenaline, which acts in seconds. Cortisol is the hormone of the hours that follow, the sustained metabolic reconfiguration that allows the body to maintain function through prolonged difficulty. Its actions are almost impossibly diverse. In the liver, it stimulates gluconeogenesis, the synthesis of new glucose from amino acids and glycerol, raising blood glucose and ensuring that the brain has the fuel it needs, regardless of dietary intake. In the muscle and connective tissue, it promotes proteolysis, the breakdown of proteins into their constituent amino acids, providing the substrates for the liver's gluconeogenesis. In the adipose tissue, it promotes lipolysis, the release of fatty acids from stored fat, providing the alternative fuel that allows muscle and other tissues to shift away from glucose, conserving it for the brain. In the immune system, cortisol suppresses inflammation and immune activation, a profound effect that is the basis of the therapeutic use of synthetic glucocorticoids in inflammatory and autoimmune conditions, and whose chronic pharmacological exploitation produces the characteristic side effects of long-term steroid therapy, weight redistribution, muscle wasting, immune suppression, thinning of the skin, osteoporosis, glucose intolerance. In the vasculature, cortisol maintains the sensitivity of blood vessels to the vasoconstrictive effects of adrenaline, the permissive effect that is why patients with adrenal insufficiency can develop dangerous hypotension even during mild stress. Cortisol is secreted in a circadian pattern, peaking in the early morning around the time of waking, and falling through the day to its nadir in the late evening, which is part of the hormonal machinery that prepares the body for the metabolic demands of the waking day and allows the metabolic quietude of sleep. The adrenal glands, two small, triangular structures sitting atop the kidneys, are actually two distinct endocrine organs fused into one, with the outer cortex producing steroid hormones under pituitary control, and the inner medulla producing catecholamines as a modified sympathetic ganglion that has been incorporated into the endocrine system. The adrenal gland is an anatomical accident of development that has turned out to be one of the body's most versatile endocrine structures. The cortex, derived from the mesoderm, and the medulla, derived from the neural crest, the same tissue that gives rise to neurons, and the chromaffin cells of the sympathetic nervous system, are embryologically unrelated, but they ended up sharing a capsule, and the result is a gland with two entirely different hormonal systems occupying the same address. The adrenal cortex is itself divided into three zones: each producing a different class of steroid hormones. The outermost zona glomerulosa produces aldosterone, the mineralocorticoid that regulates sodium reabsorption in the kidney and governs blood volume and blood pressure. The middle zona fasciculata produces cortisol, the primary glucocorticoid, under the control of ACTH from the pituitary. The innermost zona reticularis produces adrenal androgens, dehydroepiandrosterone and androstenedione, weak androgens that serve as precursors for the peripheral conversion to more potent sex steroids. All three zones use cholesterol as their starting material. The steroid hormones are lipid-derived, structurally based on the cholesterol backbone, synthesized through a series of enzymatic modifications. The adrenal medulla, in contrast, produces adrenaline and noradrenaline, not steroids but catecholamines, derived from the amino acid tyrosine, and releases them directly into the bloodstream in response to sympathetic nervous stimulation, acting as the hormonal arm of the fight or flight response. The combination of these two systems in one gland means that the adrenal gland can respond to stress on two timescales simultaneously, the medulla responding in seconds with the adrenaline surge, and the cortex responding over minutes to hours with the sustained cortisol mobilization. The parathyroid glands, for tiny glands embedded in the posterior surface of the thyroid, each approximately the size of a grain of rice, maintain calcium homeostasis with extraordinary precision through parathyroid hormone and their inadvertent removal during thyroid surgery historically produced tetany and death, revealing how essential their function is. The parathyroid glands are among the smallest endocrine organs in the body and among the most clinically consequential in their absence. Each is approximately 5 millimeters in length, weighing no more than 40 milligrams, and they sit in the loose connective tissue behind the thyroid, their positions variable enough that surgeons must actively look for them during thyroid operations. Their sole function is the maintenance of blood calcium within the narrow range of approximately 2.1 to 2.6 millimoles per liter, a range whose importance has been discussed in the context of neural function, but which is equally critical for cardiac function, for the coagulation system, for virtually every enzyme that uses calcium as a cofactor. The parathyroid chief cells contain calcium-sensing receptors, G-protein-coupled receptors that directly detect the free calcium concentration in the extracellular fluid and respond to any fall by instantly increasing the secretion of parathyroid hormone. PTH acts on 3 organs simultaneously. In the kidney, it stimulates the reabsorption of calcium from the tubular fluid, preventing its loss in the urine, and it stimulates the production of the active form of vitamin D. In bone, it stimulates osteoclast activity, mobilizing calcium from the skeleton by dissolving the hydroxyapatite mineral. The active vitamin D that PTH stimulates in the kidney then acts on the intestine to increase the absorption of dietary calcium. The three-pronged response, less calcium lost in urine, more calcium absorbed from food, more calcium released from bone, raises blood calcium back to normal in a matter of minutes. The historical catastrophe of inadvertent parathyroidectomy during early thyroid surgery, the patients developing the progressive muscular cramps and spasms of hypocalcemia within hours of surgery, sometimes dying, revealed the indispensability of a structure so small it was frequently overlooked. Aldosterone, the mineralocorticoid secreted by the adrenal zona glomerulosa in response to the renin-angiotensin-aldosterone system, is the primary regulator of sodium balance and blood volume, acting on the collecting duct of the kidney to increase sodium reabsorption and potassium excretion, and its dysregulation produces the hypertension of primary hyperaldosteronism and the hypotension of adrenal insufficiency. Aldosterone is one of the body's principal engineers of the internal sea, the extracellular fluid whose sodium concentration and volume determine blood pressure and whose stability is essential for cellular function. The renin-angiotensin-aldosterone system is the primary regulator of this fluid balance, operating as a cascading hormonal chain reaction. Falling blood volume or falling blood pressure stimulates the juxtaglomerular cells of the kidney to release renin, a protease enzyme that cleaves the plasma protein angiotensinogen to produce angiotensin I. Angiotensin I is then converted to angiotensin II by angiotensin-converting enzyme in the pulmonary circulation. Angiotensin II, a potent vasoconstrictor acting directly on blood vessels, also stimulates the adrenal zona glomerulosa to secrete aldosterone. Aldosterone enters the principal cells of the kidney collecting duct, binds to the intracellular mineralocorticoid receptor, and the hormone receptor complex migrates to the nucleus and regulates gene transcription, increasing the expression of the epithelial sodium channel at the apical membrane and of the sodium-potassium ATPase at the basolateral membrane. The result is increased sodium reabsorption from the tubular fluid into the blood, with water following osmotically, expanding the extracellular fluid volume and raising blood pressure. Potassium and hydrogen ions are excreted in exchange for the retained sodium. Aldosterone's actions are slow by hormonal standards, taking approximately 30 minutes to begin and hours to reach maximum effect, reflecting the time required for the gene regulatory effects to manifest as changes in protein expression. The pineal gland, positioned at the geometric center of the brain, synthesizes melatonin from serotonin in a reaction that requires darkness as its cue, producing the rising melatonin levels of the evening that signal the circadian system that night has arrived, and that initiate the cascade of physiological changes that prepare the body for sleep. The pineal gland's story spans from antiquity. René Descartes proposed it as the seat of the soul, the one unpaired structure in the otherwise bilaterally symmetric brain, where the immaterial mind met the material body, to the present day, where its function has been precisely characterized, and its primary product precisely identified and commercially synthesized. The gland is small, approximately 8 millimeters long, shaped roughly like a pine cone, from which it takes its name, and it sits in a cleft between the two hemispheres of the brain, posterior to the third ventricle. Its cells, the pinealocytes, receive, indirectly, information about environmental light through a pathway that runs from the retina to the suprachiasmatic nucleus of the hypothalamus, the master circadian clock, and then to the superior cervical ganglion of the sympathetic nervous system, from which noradrenergic fibers project to the pineal gland. In darkness, sympathetic activation drives the pinealocytes to convert serotonin to N-acetylserotonin, and then to melatonin through the sequential actions of ANET and HIAMT enzymes. The key regulatory step, ANET, is strongly suppressed by light and activated by noradrenaline, making the pineal essentially a darkness detector. Melatonin production begins approximately 2 hours after the onset of darkness, peaks between 2 and 4 in the morning, and falls as dawn approaches. This melatonin rhythm provides the body's circadian timing signal, the chemical announcement of night, acting on melatonin receptors in the suprachiasmatic nucleus to reinforce the circadian program, on receptors in the brain to promote sleep onset, and on receptors throughout the body to synchronize the peripheral circadian clocks that regulate the timing of virtually every physiological process. Right now, as you lie in the dark, your pineal gland is measuring the darkness and adding melatonin to your bloodstream, molecule by quiet molecule. Estrogen, actually a family of related hormones, including estradiol, estrone, and estriol, is produced primarily by the granulosa cells of the ovarian follicles in premenopausal women, and exerts its effects through nuclear receptors that regulate gene transcription in virtually every tissue in the body, making it one of the most systemically influential hormones known. Estrogen's name comes from the Greek for frenzy, which reflects the early observation of its role in driving the estrous cycle in animals, but the name undersells the breadth of its influence. The estrogen receptors, RNR, are expressed not only in the reproductive tissues, where estrogen's most obvious effects occur, but in the brain, the bone, the cardiovascular system, the liver, the skin, the gut, and effectively every tissue that has been carefully looked at. When estradiol, the most potent and predominant form of estrogen in the premenopausal years, enters a cell, it binds to its receptor in the cytoplasm or nucleus, and the hormone receptor complex acts as a transcription factor, binding to estrogen response elements in the DNA and regulating the expression of hundreds of target genes. This genomic action is slow, taking hours to manifest in changed protein expression, but it underlies estrogen's long-term effects on tissue structure and function. Estrogen promotes the deposition of bone matrix by stimulating osteoblast activity and suppressing osteoclast activity, which is why the loss of estrogen at menopause dramatically accelerates bone loss and increases fracture risk. It maintains the health and suppleness of the vaginal epithelium, the cervical mucus, the endometrium. It influences the distribution of body fat, favoring peripheral, subcutaneous, deposition over central, visceral deposition in premenopausal women. It has protective effects on the cardiovascular system, maintaining arterial elasticity, promoting favorable lipid profiles, modulating platelet function, which largely disappear at menopause, and contribute to the rising cardiovascular risk of the postmenopausal years. And it exerts profound effects on the brain, influencing mood, cognition, the regulation of temperature. The loss of its hypothalamic temperature modulating effects at menopause produces the vasomotor symptoms of hot flashes and the sleep architecture that is so frequently disrupted in the perimenopausal transition. Testosterone, the primary androgen produced by the Leydig cells of the testis in men, and in smaller amounts by the ovaries and adrenal glands in women, acts through the androgen receptor to regulate muscle protein synthesis, bone density, red blood cell production, libido, and many aspects of behavior, and its levels follow a circadian rhythm that peaks in the early morning. Testosterone is perhaps the most discussed hormone in popular culture and among the least understood in its genuine biological complexity. It is a steroid hormone derived from cholesterol through a series of enzymatic steps, and in men, its production is governed by the hypothalamic pituitary gonadal axis. The hypothalamus releases gonadotropin-releasing hormone in a pulsatile fashion, stimulating the pituitary to release LH and FSH, which act on the Leydig cells of the testis to produce testosterone in a negative feedback loop. Testosterone acts in multiple ways on its target tissues. In cells expressing the enzyme 5-alpha reductase, primarily the prostate, the skin of the face and scalp, and the external genitalia, testosterone is converted to dihydrotestosterone, a more potent androgen that is the active agent in those tissues. In other tissues, muscle, bone, the brain, testosterone itself binds to the androgen receptor. And in tissues expressing aromatase, the enzyme that converts testosterone to estradiol, found in fat tissue, the brain, the liver, and bone, testosterone serves as a precursor for local estrogen production. The relative contributions of testosterone itself, DHT, and locally produced estradiol to the male sex hormone effects on different tissues is an area of continuing investigation. The circadian rhythm of testosterone, peaking in the early morning and falling through the day, is the reason that testosterone measurements for clinical purposes are standardized to early morning blood sampling. The peak is approximately 20 to 25% above the nadir, a variation that reflects the nocturnal pulses of LH, driven by the hypothalamic GnRH rhythm during sleep. Growth hormone, released from the somatotroph cells of the anterior pituitary, in a pulsatile pattern, with the largest pulse occurring approximately an hour after the onset of deep sleep, promotes linear bone growth in childhood, stimulates protein synthesis in muscle, mobilizes fat from adipose tissue, and regulates many aspects of metabolism in adults. Growth hormone is produced throughout life, not only in childhood, but its effects shift with development in ways that reflect the changing metabolic priorities of different life stages. In childhood, its most dramatic effect is on the growth plates of the long bones, the cartilaginous epiphyseal plates where endochondral ossification progressively adds length to the skeleton. Growth hormone does not act directly on the growth plate, but stimulates the liver and other tissues to produce insulin-like growth factor 1, IGF-1, which acts on the chondrocyte of the growth plate to drive their proliferation and differentiation. The combination of the direct effects of growth hormone on many tissues and the indirect effects mediated through IGF-1 produces the systemic anabolic state of childhood growth. Protein synthesis is increased, fat is mobilized from adipose stores, and the skeleton extends. In adults whose growth plates have closed, growth hormone continues to be secreted, but its effects are primarily metabolic, maintaining lean body mass, controlling fat distribution, particularly visceral fat, preserving bone density, and governing aspects of cardiovascular function. The pulsatile pattern of growth hormone secretion, with the largest pulse occurring in the first few hours of sleep during the deep slow-wave sleep of N3, means that the quality and quantity of sleep directly influence growth hormone secretion. Children who sleep well grow better than those who sleep poorly, a relationship that is partly mediated through this nocturnal growth hormone pulse. Right now, as your body approaches deep sleep, growth hormone is beginning its nightly release, a quiet anabolic surge that will oversee the night's repair, recovery, and maintenance. Prolactin, produced by the lactotroph cells of the anterior pituitary, and uniquely regulated by tonic inhibition from dopamine, rather than stimulation, is best known for its role in initiating and maintaining lactation, but exerts over 300 distinct biological actions, making it one of the most functionally diverse hormones in vertebrate biology. The catalog of prolactin's actions across vertebrate species is one of the more remarkable lists in endocrinology. It stimulates lactation in mammals, crop milk production in pigeons, brood patch development in birds, water and electrolyte balance in fish, parental behavior in a wide range of species, immune function across taxa, and in humans, effects on metabolism, bone, the immune system, brain function, and behavior that continue to be characterized. The sheer breadth of prolactin's actions reflects its ancient evolutionary history. It is one of the oldest pituitary hormones, with structural homologs present in fish and amphibians, where it regulates osmoregulation and metamorphosis. In humans, its primary clinical significance lies in its role in lactation and its unique regulatory mechanism. Unlike the other anterior pituitary hormones, which are under positive control by hypothalamic-releasing hormones, prolactin is under tonic inhibition by dopamine, which the hypothalamus continuously releases into the portal circulation to suppress prolactin secretion. When dopamine is withdrawn, as occurs during suckling, which triggers a neural reflex that inhibits hypothalamic dopamine release, prolactin rises and maintains milk production. This dopaminergic inhibition explains why drugs that block dopamine receptors, antipsychotic medications, metoclopramide, elevate prolactin as a side effect, sometimes producing galactorrhea and menstrual irregularities in women, and gynecomastia in men. Prolactin levels also rise during sleep. Another reason why prolactin is among the hormones quietly elevated in the blood right now, as the body moves toward the deeper stages of the night. The renin-angiotensin-aldosterone system is one of the body's most important blood pressure and fluid balance regulators, operating as a multi-organ hormonal cascade that connects the kidney's sensing of perfusion pressure and sodium delivery to the adrenal gland's production of aldosterone and to the direct vasoconstrictive effects of angiotensin II throughout the circulation. The renin-angiotensin-aldosterone system is, in its architecture, a lesson in how the body constructs homeostatic systems of great sensitivity and reach, from the serial activation of enzymes and receptors across multiple organs. The kidney's juxtaglomerular apparatus, a specialized region of the distal tubule and the afferent arteriole, detects the perfusion pressure of the kidney and the sodium chloride concentration in the tubular fluid, and responds to reductions in either by secreting renin from the juxtaglomerular cells. Renin is a protease, an enzyme that cleaves other proteins, and its substrate is angiotensinogen, a large plasma protein produced continuously by the liver. Renin cleaves angiotensinogen to produce the decapeptide angiotensin I, which is then cleaved by angiotensin-converting enzyme in the pulmonary capillaries to produce the octapeptide angiotensin II. Angiotensin II acts as an extraordinarily potent vasoconstrictor, binding to at1 receptors on vascular smooth muscle cells and triggering contraction, raising peripheral vascular resistance and blood pressure directly. Simultaneously, it stimulates aldosterone secretion from the adrenal cortex. It acts on the hypothalamus to stimulate thirst and ADH secretion. It acts directly on the proximal tubule of the kidney to increase sodium reabsorption. The entire cascade, from falling blood pressure to kidney enzyme release, to liver protein to lung enzyme, to vasoconstriction and aldosterone to sodium retention, is completed within minutes and represents a distributed sensing and response system that spans 4 organs and 3 classes of hormone. Antidiuretic hormone, also called vasopressin, is released from the posterior pituitary in response to increases in plasma osmolality or decreases in blood volume, and it acts on the collecting duct of the kidney to increase water reabsorption, concentrating the urine and restoring fluid balance with a precision that is extraordinary, given the relatively crude nature of the sensing mechanism. Antidiuretic hormone is the body's principal anti-dehydration molecule. The neurons in the hypothalamic supraoptic and paraventricular nuclei that produce it are themselves osmosensors. Their firing rate increases directly in proportion to the osmolality of the cerebrospinal fluid around them, which closely reflects the osmolality of the plasma. When plasma osmolality rises, as it does during dehydration, when the ratio of solutes to water in the blood increases, the ADH neurons fire more rapidly, releasing more ADH from their terminals in the posterior pituitary into the systemic circulation. ADH travels to the kidney collecting duct, where it binds to V2 receptors on the principal cells, triggering the insertion of aquaporin 2 water channels into the apical membrane. This dramatically increases the water permeability of the collecting duct, allowing water to flow osmotically out of the tubular lumen into the concentrated medullary interstitium, concentrating the urine and retaining water for the circulation. The precision of this system is illustrated by the numbers. In the absence of ADH, the human kidney can produce up to 20 liters of dilute urine per day. In the presence of maximal ADH stimulation, it concentrates the urine to approximately 1,200 mosm/kg and produces as little as 500 milliliters per day, a 40-fold reduction in urine output. The regulation of ADH secretion by plasma osmolality is sensitive to changes of as little as 1%. The body detects a 1% rise in plasma osmolality and begins conserving water before any subjective sensation of thirst has arisen. The subjective sensation of thirst arrives at approximately 2% above baseline, by which point the physiological response has already been underway for some time. The body acts before the mind registers the problem. The gut, long regarded as simply a digestive organ, is now recognized as the largest endocrine organ in the body, producing more than 20 distinct hormones, including ghrelin, leptin, GLP-1, GIP, cholecystokinin, secretin, and peptide YY, which together coordinate digestion, appetite, insulin secretion, gastric motility, and the communication between the gut and the brain. The recognition of the gut as a major endocrine organ is one of the more significant conceptual shifts in endocrinology of recent decades. The gastrointestinal tract, from the stomach to the large intestine, is lined with enteroendocrine cells, scattered singly among the epithelial cells of the gut mucosa, in a distribution so diffuse that, if the gut's endocrine cells were collected into a single organ, that organ would be many times larger than the pancreas. These enteroendocrine cells sense the contents of the gut lumen, the presence of nutrients, the acidity, the osmolality, the specific amino acids and fatty acids and glucose that signal what has been eaten, and respond by releasing specific hormones into the portal blood or into the submucosal space, where they can act locally on the enteric nervous system. Ghrelin, produced by the oxyntic cells of the stomach fundus, is the only gut hormone that stimulates appetite. Its levels rise before meals and fall after eating, making it the stomach's hunger signal to the brain. Cholecystokinin, released from duodenal cells in response to fat and protein in the meal, stimulates the gallbladder to contract and the pancreas to release digestive enzymes, and it acts on the vagus nerve to signal satiety. GLP-1, glucagon-like peptide 1, released from L cells in the ileum and colon in response to nutrients, is the most potent gut incretin, stimulating insulin secretion from the beta cells in a glucose-dependent fashion, slowing gastric emptying, and acting on the brain to reduce appetite. Its receptor agonists have become among the most transformative drugs in the treatment of type 2 diabetes and obesity. The gut, in other words, is not merely digesting food. It is conducting a continuous chemical conversation with the brain and the pancreas about what has just been eaten and what the metabolic response should be. Leptin, the hormone produced by adipose tissue in proportion to fat mass, signals the brain about the body's long-term energy stores, suppressing appetite and increasing energy expenditure through its action on the hypothalamic arcuate nucleus. In its discovery in 1994 by Jeffrey Friedman, transformed the understanding of obesity from a failure of willpower to a disorder of hormonal signaling. The discovery of leptin, encoded by the OB gene, was preceded by a classical experiment in mouse genetics that had long suggested the existence of a circulating satiety signal. Mice with mutations in the OB gene became massively obese, eating voraciously as though perpetually starving. But when conjoined to normal mice in parabiosis experiments that allowed sharing of the blood circulation, the OB mice lost weight, as though receiving a satiety signal from the normal mouse's blood. The signal turned out to be leptin, and its identification by Friedman and his colleagues provided the first molecular understanding of how the brain knows how much fat the body is carrying. Leptin is produced by fat cells in direct proportion to their size. The more fat in the adipocytes, the more leptin they secrete. Leptin travels in the bloodstream, crosses the blood-brain barrier via a specific transporter, and acts on receptors in the hypothalamic arcuate nucleus, particularly on neurons expressing the appetite-stimulating neuropeptide Y and the appetite-suppressing POMC peptide, to suppress appetite and increase sympathetic nervous system activity and thermogenesis. In the normal feedback loop, a gain in fat mass leads to more leptin, which suppresses appetite and increases energy expenditure, returning fat mass toward its set point. In obesity, however, leptin levels are actually elevated. The fat tissue is producing abundant leptin, but the response to that leptin is blunted, a state of leptin resistance analogous to the insulin resistance of type 2 diabetes. The brain is receiving the signal that fat stores are adequate, but it is not fully responding, and appetite remains elevated. Understanding the mechanisms of leptin resistance has become a central project of obesity research. The HPA axis, the hypothalamic pituitary adrenal axis, is the body's primary stress response system, responding to physical and psychological stressors with a cascade of hormonal signals that ultimately produces the cortisol mobilization that prepares the body to deal with challenge, and whose chronic activation in the context of persistent stress produces the physiological and psychological consequences of chronic stress. The HPA axis is the endocrine translation of the nervous system's perception of threat. When the brain, through the limbic system, the cortex, or the brainstem, registers a stressor, signals converge on the paraventricular nucleus of the hypothalamus, which releases corticotropin-releasing hormone into the portal circulation of the pituitary stalk, CRH, and its synergistic partner arginine vasopressin stimulates the corticotroph cells of the anterior pituitary to secrete adrenocorticotropic hormone into the systemic circulation. ACTH acts on the adrenal cortex to stimulate the synthesis and release of cortisol within 15 to 30 minutes of the initial stress. Cortisol then implements the metabolic reconfiguration of the stress response, raising blood glucose, mobilizing energy substrates, dampening inflammation, and modulating the activity of brain regions involved in memory, attention, and emotional processing. The negative feedback of cortisol on the hypothalamus and pituitary limits the duration of the HPA response under normal circumstances. Rising cortisol suppresses further CRH and ACTH release, ensuring that the stress response is self-limiting. But in the context of chronic, unresolvable stress, the kind that modern life increasingly produces, the normal feedback mechanisms are overwhelmed, and the HPA axis remains chronically elevated, producing sustained high cortisol levels. Chronic HPA activation produces predictable and serious physiological consequences: immune suppression, increased cardiovascular risk, disrupted sleep, impaired memory, reduced bone density, and the dysregulation of metabolic processes. The HPA axis is designed for acute challenges that resolve. It is poorly adapted for the prolonged, inescapable stressors of the contemporary world. Oxytocin, the nonapeptide hormone synthesized in the hypothalamus and released from the posterior pituitary, is most famous for its roles in childbirth and breastfeeding, but is increasingly understood to be a broad social bonding hormone that modulates trust, empathy, anxiety, and the neural processing of social information throughout the lifespan. Oxytocin, from the Greek for swift birth, was first characterized for its role in stimulating uterine contractions during labor, a function exploited clinically when synthetic oxytocin is administered to induce or augment labor. It was subsequently found to be the hormone responsible for the milk ejection reflex during breastfeeding, the letdown that occurs when the suckling infant triggers a neuroendocrine reflex that releases oxytocin from the posterior pituitary, causing the myoepithelial cells surrounding the milk-producing alveoli of the mammary gland to contract and expel milk. These two functions, uterine contraction and milk ejection, were the classical picture of oxytocin's role. The broader social and behavioral functions of oxytocin began to emerge from comparative studies, particularly in foals, that revealed oxytocin's role in pair bonding and parental behavior, and subsequently from human studies showing that intranasal oxytocin administration altered social cognition, increased trust in economic games, enhanced the recognition of emotional facial expressions, and modulated social anxiety. The mechanism appears to involve oxytocin's action in the amygdala and the mesolimbic dopamine system, reducing the amygdala's threat response to social stimuli and enhancing the reward system's response to social reward. making social interactions simultaneously less threatening and more pleasurable. Oxytocin is released during social touch, eye contact, positive social interactions, and orgasm, and its release in one person tends to stimulate its release in the person they are interacting with, creating a mutual biochemical facilitation of social bonding. The hormone of childbirth and breastfeeding turns out to be, more broadly, the hormone of closeness. The thyroid hormones cross the placenta to the developing fetus and are essential for normal fetal brain development, particularly the maturation of neurons and myelination of axons in the second trimester. An iodine deficiency during pregnancy, which impairs maternal thyroid hormone production, remains the most preventable cause of intellectual disability worldwide. The fetal brain's dependence on maternal thyroid hormones is one of the most consequential facts in developmental endocrinology, and it reveals a vulnerability in human development that has profound public health implications. The fetal thyroid gland does not begin producing its own thyroid hormones in meaningful quantities until approximately week 18 to 20 of gestation. Before this point, and to a significant degree after it as well, the fetal brain depends on maternal thyroid hormones crossing the placenta to drive its development. During the critical period of brain development in the first and second trimesters, thyroid hormones regulate the proliferation and migration of neurons, the formation of synaptic connections, and the myelination of axons, processes that cannot be adequately compensated if thyroid hormone is deficient. Iodine is required for the synthesis of thyroid hormones. In populations in areas of the world where soil and water iodine content is low, mountainous regions far from the sea, where iodine-depleted glacial and rainwater has leached the mineral from the soil, have historically had high rates of iodine deficiency goiter, and of the intellectual disability associated with severe fetal hypothyroidism, a condition once called cretinism. The global public health campaign to iodize salt, begun in the early 20th century and now covering much of the world's population, has been one of the most successful micronutrient interventions in history, dramatically reducing the prevalence of iodine deficiency and its consequences. Adequate iodine intake during pregnancy, slightly higher than the non-pregnant requirement, is one of the most important nutritional factors for fetal neurodevelopment. A hormone whose synthesis requires a dietary mineral is one of the most powerful shapers of the human brain. The gonadotropins, follicle-stimulating hormone and luteinizing hormone, both produced by the gonadotroph cells of the anterior pituitary, drive the development of the gametes, regulate the production of sex hormones from the gonads, and in women, the precisely timed surge of LH at mid-cycle is the hormonal trigger of ovulation. FSH and LH are both glycoprotein hormones, proteins with carbohydrate chains attached, and both bind to G-protein-coupled receptors on their target cells in the gonads, triggering the CAMP signaling cascade. Their pulsatile release from the pituitary is driven by the pulsatile release of GnRH from the hypothalamus, a pattern of secretion whose frequency and amplitude changes dramatically across the menstrual cycle, and whose disruption, by excessive exercise, extreme caloric restriction, or stress, suppresses gonadotropin secretion and disrupts reproductive cycling. In women, FSH drives the development of ovarian follicles in the early follicular phase of the menstrual cycle, selecting the cohort of follicles that will grow, of which usually one will become the dominant follicle. The growing follicles produce estradiol, which rises through the follicular phase and exerts a negative feedback effect on FSH secretion, limiting the number of follicles that mature to one. The rising estradiol also, at sufficiently high levels sustained for long enough, switches from negative to positive feedback on the pituitary, triggering the LH surge, a dramatic, brief elevation in LH secretion that peaks approximately 12 to 16 hours before ovulation and triggers the final maturation and rupture of the dominant follicle, releasing the oocyte. The transition from negative to positive feedback of estradiol on the pituitary, unique among hormonal feedback systems in its direction reversal, is one of the most precisely timed molecular events in reproductive physiology, and its dependence on both the level and the duration of the estradiol exposure explains why the timing of the LH surge is so precisely coordinated with the development of the dominant follicle. Insulin resistance, the reduced responsiveness of target tissues to insulin signaling, requiring greater concentrations of insulin to produce the same degree of glucose uptake, is the central defect of type 2 diabetes and metabolic syndrome, and its development is driven by a complex interaction of obesity, physical inactivity, inflammation, and genetic susceptibility. Insulin resistance is not a single molecular lesion, but a heterogeneous failure of multiple steps in the insulin signaling pathway across different tissues, with different predominant defects in different tissues in different people. The insulin receptor is a transmembrane tyrosine kinase. When insulin binds to its extracellular domain, the receptor autophosphorylates and initiates a signaling cascade through the IRS proteins, PI3 kinase, and ACT, that ultimately drives the translocation of GLUT4 glucose transporters to the cell surface in muscle and fat cells, and regulates glucose and lipid metabolism in the liver. In insulin resistance, this cascade is impaired at one or more steps. The receptor may not phosphorylate efficiently. The IRS proteins may be degraded by chronic inflammatory signaling. The PI3 kinase pathway may be attenuated. The result is that a given concentration of insulin produces less glucose uptake than normal. The pancreatic beta cells compensate by secreting more insulin, hyperinsulinemia, which maintains blood glucose in the normal range for years, concealing the underlying resistance. Eventually, if the beta cells exhaust their compensatory capacity, as they do in a fraction of people with insulin resistance, particularly those with a genetic predisposition to beta cell failure, blood glucose rises, and type 2 diabetes is diagnosed. The metabolic consequences of chronic insulin resistance and hyperinsulinemia, before and independent of diabetes, include dyslipidemia, hypertension, systemic inflammation, and increased cardiovascular risk, the cluster of abnormalities called metabolic syndrome. Insulin resistance is the central hormonal nexus through which obesity, inactivity, and genetic susceptibility converge to produce a cascade of chronic metabolic disease. Calcitonin, produced by the parafollicular C cells of the thyroid gland in response to elevated blood calcium, acts on bone osteoclasts to inhibit bone resorption, lowering blood calcium, and serves as the functional counterpart to parathyroid hormone in calcium homeostasis, though its physiological importance in humans appears to be modest compared to its dramatic pharmacological effects at high doses. Calcitonin occupies an unusual position in endocrinology. It is a hormone with clear physiological effects in experimental settings and at pharmacological doses, whose role in the normal day-to-day regulation of calcium homeostasis in humans appears to be relatively minor, despite its theoretical importance as the opponent of parathyroid hormone in the calcium regulatory system. The evidence for its modest physiological role comes from clinical observation, removal of the thyroid, and therefore the C cells that produce calcitonin, does not reliably produce hypercalcemia, suggesting that the body can regulate calcium adequately without calcitonin. Calcitonin's physiological importance may be greatest during periods of calcium stress, pregnancy and lactation, when the demands on the skeleton are highest, where it may protect the maternal skeleton from excessive resorption. Its pharmacological importance, however, is considerable at the concentrations used therapeutically. Calcitonin is a potent inhibitor of osteoclast activity, reducing bone pain in Paget's disease, lowering the hypercalcemia of malignancy, and transiently reducing the bone resorption of osteoporosis. Calcitonin is a 32 amino acid peptide whose structure is highly conserved across vertebrates, suggesting an important evolutionary role. In the salmon version of the hormone, salcitonin is considerably more potent at human receptors than the human version, a species difference that has been extensively exploited in pharmacology. The adrenal androgens, dehydroepiandrosterone and androstenedione, produced by the adrenal zona reticularis, serve as the principal sex steroid precursors in women, where peripheral conversion of these weak androgens to estradiol and testosterone accounts for a substantial fraction of total sex steroid production, particularly after menopause when ovarian production has ceased. The adrenal gland's production of sex steroid precursors represents one of the hormonal system's more elegant redundancies, a backup source of sex steroids that becomes particularly important in situations where the primary sources are unavailable. In premenopausal women, the ovaries are the dominant source of estradiol, but the adrenal zona reticularis continuously produces DHEA and androstenedione, androgens that, while biologically weak in themselves, are converted to more potent sex steroids in peripheral tissues expressing the relevant enzymes. Aromatase in fat tissue, breast tissue, and bone converts androstenedione to estrone, and DHEA to estradiol. In postmenopausal women, where the ovaries have ceased estrogen production, peripheral aromatization of adrenal androgens becomes the dominant source of circulating estrogen, which is why obesity, with its increased mass of aromatase-containing fat tissue, is associated with higher postmenopausal estrogen levels. This same mechanism underlies the increased risk of estrogen receptor-positive breast cancer in postmenopausal obese women, whose elevated circulating estrogen, derived from adrenal precursors aromatized in fat, drives the proliferation of estrogen-sensitive breast cells. DHEA is the most abundant adrenal steroid by mass and circulates at much higher concentrations than cortisol or aldosterone, but its biological role remains somewhat uncertain. Its serum levels are high in young adults and decline progressively with age, and the significance of this age-related decline for health has been extensively studied and remains contested. Erythropoietin, the glycoprotein hormone produced predominantly by the peritubular cells of the kidney in response to hypoxia, stimulates the bone marrow to increase red blood cell production, maintaining the oxygen-carrying capacity of the blood and constituting one of the clearest examples of a closed-loop endocrine feedback system. Erythropoietin, EPO, is the body's oxygen delivery optimizer, a hormone whose sole purpose is to ensure that the blood's red blood cell mass is calibrated to the oxygen demands of the tissues. The sensing mechanism is elegant. The peritubular fibroblasts of the kidney contain HIF proteins, hypoxia-inducible factors, that are normally rapidly degraded in the presence of adequate oxygen, but stabilize and accumulate when oxygen supply falls. Stabilized HIF-1 and HIF-2 act as transcription factors, driving the expression of the EPO gene and increasing erythropoietin secretion. The secreted EPO travels via the bloodstream to the bone marrow, where it binds to receptors on erythroid progenitor cells, preventing their programmed cell death and driving their proliferation and differentiation into mature red blood cells. Over days to weeks, the increased marrow output raises the circulating red blood cell count, increasing the blood's oxygen-carrying capacity and restoring the tissue oxygen delivery that had triggered the EPO signal. As oxygen delivery normalizes, HIF proteins are again degraded, EPO secretion falls back to basal levels, and the erythroid stimulus is withdrawn. The feedback is entirely oxygen-mediated. The sensing is of oxygen. The response increases oxygen delivery, and the restoration of adequate oxygen delivery extinguishes the response. The clinical exploitation of this system has created both the therapeutic use of recombinant EPO and the anemia of chronic kidney disease, where the damaged kidney cannot produce sufficient EPO, and the notorious doping of high-altitude training and synthetic EPO administration in endurance sports, where artificially elevated hematocrit provides a performance advantage by increasing the oxygen delivery to working muscles. The menstrual cycle. The approximately 28-day hormonal cycle governing the preparation and shedding of the uterine endometrium is orchestrated by the precisely timed interactions of four hormones: estradiol, progesterone, FSH, and LH, in a feedback system whose complexity, reproducibility, and sensitivity to disruption reveals the extraordinary precision required to maintain reproductive cycling. The menstrual cycle is a masterpiece of hormonal choreography, requiring the precise coordination of hormone secretion across the hypothalamus, the pituitary, the ovary, and the uterus in a sequence that must be reproduced approximately 500 times in a lifetime, with the right hormones rising and falling in the right order, at the right rates, reaching the right peak concentrations for the right durations, to maintain reproductive function. The cycle begins at the first day of menstruation, the shedding of the endometrial lining, when estradiol and progesterone are at their lowest, and FSH is beginning to rise as the feedback suppression of the previous cycle wanes. Rising FSH drives the development of a new cohort of follicles, whose granulosa cells produce estradiol in rising amounts as the follicles grow. Through the follicular phase, rising estradiol suppresses FSH, limiting the follicle cohort to a single dominant follicle, while building the uterine endometrium and priming the pituitary for the LH surge. At approximately day 13 or 14, the high estradiol triggers the LH surge. Ovulation releases the oocyte, and the ruptured follicle becomes the corpus luteum. The corpus luteum produces progesterone, which transforms the proliferative endometrium into a secretory one, receptive to implantation, along with estradiol. If no pregnancy occurs, the corpus luteum involutes after approximately 14 days. Progesterone and estradiol fall, the endometrium sheds, and the cycle begins again. The cycle's sensitivity to disruption, by stress, by changes in body weight, by illness, by excessive exercise, reflects the fact that the hypothalamic GnRH pulse generator that drives the whole system is itself modulated by energy availability, cortisol, leptin, and many other signals that communicate the body's overall state to the reproductive axis. Insulin-like growth factor 1, IGF-1, produced primarily by the liver, in response to growth hormone, is the primary mediator of growth hormone's effects on linear bone growth and protein anabolism, and its serum levels serve as the best clinical marker of growth hormone action, being more stable across the day than the pulsatile growth hormone itself. IGF-1 was originally called somatomedin C, a somatotropin mediator, emphasizing its role as the intermediary between growth hormone and its effects on peripheral tissues. It is a single-chain, 70 amino acid protein with significant structural homology to insulin, and like insulin, it acts through a tyrosine kinase receptor, though distinct from the insulin receptor, triggering an overlapping but distinguishable set of downstream signaling events. The primary IGF-1 effector pathway for growth is through the PI3K-ACT-mTOR cascade, which promotes protein synthesis, cell proliferation, and cell survival. In the growth plate, the cartilaginous epiphyseal plate of the developing long bone, IGF-1 acts on the chondrocyte to drive their proliferation and column formation, and on the osteoblasts, invading the mineralizing cartilage to drive bone matrix synthesis. The combination of growth hormone, acting directly on some tissues, and IGF-1 mediating its effects on others, produces the systemic anabolic state of normal childhood growth. IGF-1 stability in the blood. It circulates bound to binding proteins, particularly IGF-BP3, which slow its clearance and dampen the fluctuations, makes it a more practical clinical measurement than growth hormone, which disappears from the blood rapidly between pulses and shows enormous within-day variability. Measuring IGF-1 provides an integrated assessment of growth hormone secretion over the preceding day or two, and it is the primary laboratory test used in the diagnosis of growth hormone deficiency and acromegaly, the syndrome of excess growth hormone in adults, in which IGF-1 is markedly elevated, alongside the characteristic somatic changes of enlarged hands and feet, thickened facial features, and increased visceral organ size. Progesterone, the steroid hormone produced primarily by the corpus luteum after ovulation and by the placenta during pregnancy, acts on the uterine endometrium to prepare it for implantation, suppresses further ovulation during the luteal phase in pregnancy, and exerts sedative and anxiolytic effects on the brain through its metabolite allopregnanolone, which potentiates GABA-A receptors. Progesterone's name, from the Latin for promoting gestation, accurately captures its central reproductive function, but its neurological effects are among the more surprising and clinically relevant aspects of its biology. The most potent neuroactive metabolite of progesterone is allopregnanolone, a neurosteroid that is produced from progesterone in the brain and in peripheral tissues, and that acts as a positive allosteric modulator of GABA-A receptors, the same receptors targeted by benzodiazepines and alcohol, whose activation produces sedation, anxiolysis, and anticonvulsant effects. The sedative and calming quality that many women report during the luteal phase of the menstrual cycle, when progesterone and allopregnanolone levels are highest, reflects this direct neurological action. Conversely, the rapid fall in progesterone and allopregnanolone levels in the days before menstruation and the dramatic fall following delivery has been proposed as a mechanism for premenstrual mood changes and for postpartum depression, respectively. A synthetic analog of allopregnanolone, brexanolone, has been approved for the treatment of postpartum depression, providing the neurosteroid that is lost with the fall in progesterone. Progesterone's thermogenic effect, raising basal body temperature by approximately 0.5 degrees Celsius in the luteal phase, provides the basis for the temperature-based methods of fertility tracking. And its endometrial effects, transforming the proliferative, estrogen-stimulated endometrium into the secretory, progesterone-dominated endometrium of the luteal phase, make it the essential hormone of implantation, without which a fertilized egg cannot attach and develop. You have been with me for a while now, traveling through the chemistry of the body's long game. The hormones that run in your blood right now, that have been shaping you quietly since before you were born. I find that genuinely wonderful to think about. If these nights together mean something to you, if there is something in the pace of this, in the way the science of the body becomes a kind of rest for the mind, then subscribing to Relax with Science is a simple and warm way to say, yes, I would like more of this. I would like to come back. And I will be here whenever you do. The placenta, the temporary endocrine organ formed during pregnancy, produces human chorionic gonadotropin, progesterone, estrogen, human placental lactogen, and many other hormones, and its establishment as a functioning endocrine gland within the first weeks of pregnancy is essential for maintaining the pregnancy until the ovarian corpus luteum eventually relinquishes hormonal control. The placenta is the most extraordinary, temporary endocrine organ the body produces, a structure that assembles itself from the fusion of fetal trophoblast cells with the maternal decidua, establishes a hormonal production program within days of implantation, and then, having served its purpose, is expelled at birth and never appears again. Its first and most urgent hormonal task is the prevention of the corpus luteum's normal involution. Within days of implantation, the trophoblast cells begin producing human chorionic gonadotropin, HCG, the hormone detected by pregnancy tests, which binds to LH receptors on the corpus luteum and rescues it from the programmed death that would otherwise occur approximately 14 days after ovulation. The corpus luteum, sustained by HCG, continues to produce the progesterone and estradiol that maintain the endometrium. As the first trimester progresses, the placenta itself develops the capacity to produce progesterone directly from maternal cholesterol, the luteoplacental shift, and by approximately 8 weeks, the placenta is producing enough progesterone to sustain the pregnancy, independently of the corpus luteum. HCG peaks between 8 and 10 weeks of pregnancy, and then declines, the fall in HCG correlating, in many women, with the improvement in first trimester nausea that tends to occur in the second trimester. Through the remainder of pregnancy, the placenta produces rising levels of estrogen and progesterone, human placental lactogen, which promotes insulin resistance in the mother, diverting nutrients toward the fetus, and many other signals that coordinate the maternal adaptations to pregnancy. The adipose tissue, long considered a passive energy store, is now recognized as an active endocrine organ, producing a range of hormones called adipokines, including leptin, adiponectin, resistin, and visfitin, which regulate metabolism, insulin sensitivity, inflammation, and appetite in ways that connect body fat directly to metabolic disease risk. The endocrine reclassification of fat tissue is one of the more consequential conceptual shifts in metabolism research. The discovery of leptin in 1994, produced by fat cells and signaling the brain about energy stores, was the first clear evidence that adipose tissue was an endocrine organ. Adiponectin followed, a hormone produced exclusively by adipocytes, that increases insulin sensitivity in the liver and muscle, promotes fatty acid oxidation, and has anti-inflammatory effects. Adiponectin levels are paradoxically reduced in obesity, despite the increased adipose tissue mass, and the degree of adiponectin deficiency correlates with insulin resistance and metabolic disease risk. This reduction appears to reflect the chronic low-grade inflammation of adipose tissue in obesity, which suppresses adiponectin secretion from the adipocytes. Resistin, identified in mice as a hormone that reduced insulin sensitivity with higher levels in obesity, has a more complex and still somewhat contested role in human biology. Visfitin, produced primarily by visceral fat, has insulin mimetic properties and is elevated in obesity and metabolic syndrome. The visceral adipose tissue, the fat surrounding the abdominal organs, is more endocrinologically active and more metabolically harmful than the subcutaneous fat of the periphery, partly because its adipokine profile is more inflammatory, and partly because its portal drainage delivers its secretions directly to the liver. The distribution of body fat, how much is visceral versus subcutaneous, is consequently more important for metabolic risk than total fat mass alone, and the waist circumference, which reflects visceral fat, is a better predictor of metabolic disease risk than body mass index. Cortisol follows a precise circadian rhythm, with serum levels rising sharply in the early morning hours, peaking approximately 30 to 45 minutes after waking in the cortisol awakening response, and declining through the day to their lowest levels in the late evening. And this rhythm is driven by the suprachiasmatic nucleus clock through the HPA axis, and is essential for the biological preparation of the body for the demands of the waking day. The cortisol awakening response, the sharp rise in cortisol in the first 30 to 45 minutes after waking, producing a cortisol level 2 to 3 times the pre-wake baseline, is one of the most reliably measured hormonal responses in circadian biology, and one of the most diagnostically informative. It is not simply the cortisol level waking up with the person. It begins before waking, driven by the anticipatory activation of the HPA axis by the suprachiasmatic nucleus, and it is amplified by the act of waking and by the signals that anticipate the transition from sleep to waking activity. The cortisol awakening response is a biological preparation. The rising cortisol activates metabolic processes, increases blood glucose availability, raises alertness, modulates immune function, and prepares the brain and body for the demands of the waking day. People with more demanding anticipated days show larger cortisol awakening responses. The anticipation of challenge is itself enough to activate the HPA axis. The cortisol awakening response is also a sensitive marker of HPA axis function. Blunted responses are found in burnout, chronic fatigue syndrome, and atypical depression, exaggerated responses in people under high anticipatory stress. The circadian decline of cortisol through the day, supported by the rising melatonin of the evening, is part of the hormonal scaffolding of the sleep-wake cycle. The low cortisol evening environment in which you are now is the permissive condition for melatonin's rise, for the hypothalamic sleep centers to become active, and for the transition to the restorative biochemistry of the night. The feedback loops that regulate hormonal systems. Negative feedback, in which the product of a hormonal cascade inhibits the signals that initiated it, and the rarer positive feedback, in which the product amplifies the initiating signal, are the fundamental control mechanisms that give the endocrine system its remarkable stability and precision. The negative feedback loop is the engineering principle underlying almost every hormonal axis in the body, and understanding it is understanding the essential logic of endocrine regulation. Consider the thyroid axis. The hypothalamus releases TRH, which stimulates the pituitary to release TSH, which stimulates the thyroid to produce T3 and T4. T3 and T4, rising in the blood, feed back to both the hypothalamus and the pituitary, and suppress the release of TRH and TSH, the product of the cascade, inhibiting the signals that produced it. If thyroid hormone levels fall, as in hypothyroidism, the negative feedback is reduced. TRH and TSH rise in compensation, attempting to drive the thyroid to produce more hormone. If thyroid hormone levels rise excessively, as in hyperthyroidism, the negative feedback is increased. TRH and TSH are suppressed. This is why measuring TSH is such an informative test of thyroid function. An elevated TSH tells you that the pituitary is seeing insufficient thyroid hormone. A suppressed TSH tells you it is seeing too much. The feedback loop uses the pituitary as a sensor that can be read from the outside. Positive feedback, the system in which the product amplifies the initiating signal rather than suppressing it, is rare in endocrinology precisely because it is inherently unstable, tending toward runaway amplification. The major example is the LH surge, high estradiol, rather than suppressing the pituitary's LH secretion, as it does at lower concentrations, stimulates a massive LH release, the surge that triggers ovulation. This positive feedback is necessarily brief, self-limiting by virtue of the ovulation it triggers. Thyroxine is converted to the biologically active triiodothyronine in peripheral tissues by a family of deiodinase enzymes, whose activity varies between tissues and in response to physiological state, allowing the body to regulate the amount of active thyroid hormone available to different tissues, independently of the thyroid gland's output. The conventional picture of thyroid hormone action: the thyroid produces T4, releases it into the bloodstream, and T4 acts on target tissues, is a simplification that misses what is, in many respects, the more important level of thyroid hormone regulation: the peripheral conversion of the relatively inactive T4 to the more active T3 by tissue-specific deiodinase enzymes. Type 1 deiodinase, expressed primarily in the liver and kidney, is the major source of circulating T3, Its activity determines the systemic availability of T3 from circulating T4. Type 2 deiodinase, expressed in the brain, pituitary, and brown adipose tissue, produces T3 locally within these tissues. From locally taken up T4, these tissues can regulate their own T3 availability, independently of circulating T3 levels. Type 3 deiodinase inactivates both T4 and T3 by removing iodine from a different position, producing the biologically inactive reverse T3 and the biologically inactive T2, a local tissue-specific mechanism for switching off thyroid hormone action. The relevance of this deiodinase system becomes clear in several physiological states. During fasting and illness, type 3 deiodinase activity increases in many tissues, while type 1 activity falls, shifting the balance from T3 production to reverse T3 production, reducing the metabolic rate as a conservation mechanism. This is the euthyroid 6 syndrome. Normal thyroid gland function, normal circulating T4, but reduced circulating T3 and elevated reverse T3, as the body downregulates its own metabolic rate in response to the energy stress of illness. The thyroid gland is, in this system, one level of control among several. Steroid hormones, including cortisol, aldosterone, the sex steroids, and vitamin D, are synthesized from cholesterol through a series of enzymatic steps in the mitochondria and endoplasmic reticulum of steroidogenic cells, and because they are lipid-soluble, they diffuse freely through cell membranes to act on intracellular receptors that regulate gene transcription. The shared cholesterol origin of the steroid hormones is both a biochemical curiosity and a clinical reality. It explains why the pathway to each major steroid can be rerouted when enzymes are deficient or hyperactive, producing the characteristic hormonal patterns of the congenital adrenal hyperplasias. The first and rate-limiting step in steroidogenesis, the conversion of cholesterol to pregnenolone by the cytochrome P450 enzyme CYP11A1 in the inner mitochondrial membrane, is the committed step, stimulated by the tropic hormones, ACTH for the adrenal cortex, LH for the Leydig cells and the theca cells of the ovary, and FSH for the granulosa cells. Pregnenolone is then progressively modified by a series of hydroxylations, oxidations, and isomerizations into the major steroid hormones, the specific pathway taken, determined by which downstream enzymes are expressed in each steroidogenic cell type. The zona glomerulosa lacks the enzyme CYP17A1, but expresses CYP11B2, directing pregnenolone toward aldosterone. The zona fasciculata expresses CYP17A1 and CYP11B1, directing the pathway toward cortisol. The gonads express CYP17A1 and aromatase, directing the pathway toward the sex steroids. The lipid solubility of the steroid hormones means that they do not require membrane receptors or second messenger systems. They diffuse directly through the cell membrane, and bind to nuclear receptors in the cytoplasm or nucleus, where the hormone receptor complex acts as a transcription factor, binding to specific hormone response elements in the DNA and regulating gene expression. This genomic mechanism is slow, producing effects over hours to days, but it allows steroid hormones to fundamentally reprogram the protein expression profile of their target cells. The thyroid gland's production of hormones requires iodine in amounts that most global populations historically found difficult to obtain reliably. In the goiter, the visible enlargement of the thyroid was so common in iodine-deficient inland regions that it was once considered a normal feature of human appearance in many parts of the world. Before the understanding of iodine's role in thyroid function, and before the introduction of iodized salt, goiter was a remarkably common condition in populations living far from the sea, where iodine, which enters the food chain primarily through seafood and through the uptake of soil iodine into crops and dairy, was chronically deficient. The thyroid's response to iodine deficiency is to enlarge the TSH-driven hyperplasia of the follicular cells, attempting to extract more iodine from the meager supply and to compensate for the reduced hormone production per cell by increasing the number of cells. The result, the visible swelling at the base of the neck, that can range from a small nodularity to an enormous mass, capable of compressing the trachea and esophagus, was so prevalent in the Alps, the Himalayan foothills, the Great Lakes region of North America, and many other inland areas, that it was described in historical texts as a regional normality. Alexander the Great is reported to have noted goiter among the populations of Central Asia. Leonardo da Vinci drew goitrous necks in his anatomical illustrations. In some populations, the goiter rate exceeded 50% of adults. The introduction of iodized salt, first implemented in Switzerland and the United States in the early 1920s, is one of the great triumphs of preventive medicine, virtually eliminating endemic goiter in populations where it has been consistently applied at a cost of less than a cent per person per year. The concept of hormonal half-life, the time required for the blood concentration of a hormone to fall by half, determines the duration of hormonal signaling and the dosing interval for hormone replacement therapies, with peptide hormones typically having half-lives of minutes and steroid hormones of hours to days. Hormonal half-life is not merely a pharmacological convenience. It is a fundamental property that shapes the character of hormonal signaling. A hormone with a half-life of minutes, like insulin, growth hormone, or many of the gut peptides, can produce rapid, sharply defined pulses of activity that turn on and off quickly. A hormone with a half-life of hours, like cortisol, produces sustained, slowly changing effects that outlast the stimulus that produced them. A hormone with a half-life of days, like thyroxine, whose half-life is approximately 7 days, produces effects that are essentially constant over the short term, changing only gradually as production rates change. The differences in half-life reflect the different modes of hormone elimination. Peptide hormones are cleared from the circulation, primarily by proteolytic degradation. Circulating proteases cleave the peptide bonds, and the resulting fragments are cleared by the kidney. This proteolytic degradation is rapid, and the half-life is accordingly short. Steroid hormones are cleared primarily by hepatic metabolism. The liver attaches polar groups to the lipophilic steroid, increasing its water solubility and allowing renal excretion. This process is slower, producing longer half-lives. Thyroid hormones are highly protein-bound in the circulation. Only a tiny fraction is free and biologically active, and the protein-bound fraction acts as a reservoir that sustains the free fraction. The very high degree of protein binding of T4, approximately 99.9% bound, is the primary reason for its long half-life. The clinical implications of half-life are everywhere in endocrine medicine. The once daily dosing of thyroxine versus the multiple daily dosing of short-acting insulin. The difficulty of precisely mimicking the pulsatile secretion of natural hormones with exogenous preparations. The persistence of steroid side effects long after the drug has been discontinued. Ghrelin, the 28 amino acid peptide produced by oxyntic cells of the stomach, is the only known circulating hormone that increases appetite. In its unique orexigenic action, pulsatile pattern before meals, and interaction with the growth hormone secretagogue receptor, make it a molecule of central importance in understanding hunger, meal initiation, and the body weight set point. Ghrelin occupies a distinctive place in the hormonal regulation of appetite. It is the only peripheral hormone that unambiguously increases hunger rather than suppressing it. All of the gut-derived satiety signals, cholecystokinin, GLP-1, peptide YY, work in the direction of reducing appetite and meal size. Ghrelin works in the opposite direction, rising in the hours before a meal, reaching its peak just before eating, and falling sharply within an hour of food consumption. Its primary action is on the hypothalamic arcuate nucleus, where it activates the neuropeptide Y and AGRP neurons that drive appetite and inhibit energy expenditure. Ghrelin also acts on the pituitary to stimulate growth hormone release, which is how its receptor was first characterized as the growth hormone secretagogue receptor before ghrelin itself was identified. The pulsatile pattern of ghrelin secretion before meals, which appears to anticipate habitual meal times, suggesting that ghrelin pulsatility is partly conditioned by eating patterns, has led to the hypothesis that ghrelin drives the timing of meal initiation, as well as its strength. In the context of weight loss, ghrelin's behavior is important and sometimes frustrating. Following intentional weight loss, ghrelin levels tend to rise and remain elevated for extended periods, contributing to the persistent hunger that makes weight maintenance difficult. The body's hormonal response to weight loss, elevated ghrelin, reduced leptin, is a defense of the prior weight, a hormonal drive toward the previous energy stores. Adiponectin, the protein hormone produced exclusively by adipocytes and circulating at extremely high concentrations in the blood, compared to other hormones, has anti-diabetic, anti-inflammatory, and anti-atherogenic properties, and its paradoxical decline in obesity, despite increased fat mass, is a central feature of the hormonal dysfunction that accompanies excess adiposity. Adiponectin is unusual among hormones in its extraordinarily high circulating concentration. Measured in micrograms per milliliter, it circulates at concentrations several orders of magnitude higher than most hormones. It is the most abundant adipokine by mass, and its physiological importance is underscored by the fact that its absence in adiponectin knockout mice produces insulin resistance and metabolic syndrome, while its overexpression is protective. Adiponectin acts primarily on the liver and skeletal muscle through two receptors, ADPOR1, predominantly in skeletal muscle, and ADPOR2, predominantly in the liver, and through a pathway involving AMPK activation and the increase of fatty acid oxidation. It improves insulin sensitivity in both organs, reduces hepatic glucose production, and increases glucose uptake in muscle. Its anti-inflammatory effects, suppressing the NF-κB pathway in macrophages and endothelial cells, reducing the production of pro-inflammatory cytokines, and inhibiting the foam cell transformation that initiates atherosclerotic plaques, explain its anti-atherogenic properties. The paradox of adiponectin's decline in obesity, despite the fact that fat tissue is its source, reflects the inflammatory state of the enlarged adipocytes and the adipose tissue macrophages in obesity, which suppress adiponectin gene expression. Visceral fat, the metabolically most active and most inflammatory fat depot, is the most adiponectin deficient, which is part of the reason why central obesity is more metabolically harmful than peripheral obesity. Before we go any further, come back to the body for a moment. Feel where you are right now. The warmth of the bed, or the chair, or whatever surface is receiving you. The weight of the limbs. The rhythm of the breath, which has been slower, perhaps, than it was when you first arrived here tonight. The temperature of the skin that is exposed to the air. All of this, the warmth, the weight, the breath, the chemical balance of the blood, is being quietly governed, right now, by the hormonal system you have been hearing about. The melatonin is rising. The cortisol is low. The body knows what time it is. Let it know. Let it do what it knows how to do. Insulin secretion is not a simple, glucose concentration dependent switch, but a complex, multiphasic process involving two distinct waves of insulin release. An immediate first phase, lasting 1 to 3 minutes, and a sustained second phase, lasting as long as the glucose stimulus persists, mediated by different pools of insulin-containing vesicles within the beta cell. the dynamics of insulin secretion from the pancreatic beta cell, the timing and pattern of release in response to glucose are critically important for the normal regulation of postprandial blood glucose, and their disruption is an early marker of beta cell dysfunction in the development of type 2 diabetes. When blood glucose rises after a meal, glucose enters the beta cell through GLUT2 transporters and is metabolized, raising the ATP to ADP ratio. This rise in ATP closes ATP-sensitive potassium channels, depolarizing the cell membrane and opening voltage-gated calcium channels. The resulting calcium influx triggers exocytosis of insulin-containing secretory granules, the first phase of secretion. This first phase draws from a small pool of granules already docked at the plasma membrane, the readily releasable pool, and depletes it within minutes, producing the sharp initial spike of insulin that appears in the portal blood within 1 to 2 minutes of a glucose stimulus. The first phase primes the liver, which receives this portal insulin before the peripheral tissues to suppress hepatic glucose production. The second phase of insulin secretion is slower and more sustained, drawing from a larger reserve pool of granules that must first be recruited and primed for exocytosis, and it maintains the insulin stimulus for as long as glucose remains elevated. In people developing type 2 diabetes, the first phase of insulin secretion is specifically and selectively lost before other aspects of beta cell function deteriorate, making it both a sensitive early marker and a pathophysiologically important defect. The loss of the sharp initial insulin spike delays hepatic glucose suppression and contributes to the postprandial glucose excursions of early diabetes. Vitamin D, technically a prohormone rather than a vitamin, since the body synthesizes it from cholesterol under ultraviolet light, undergoes sequential hydroxylation in the liver and kidney to produce the active hormone calcitriol, which acts through nuclear vitamin D receptors, expressed in virtually every tissue to regulate calcium absorption, immune function, cell differentiation, and gene expression. The reclassification of vitamin D from a vitamin to a prohormone reflects a growing understanding of its scope of action that extends far beyond the dietary supplement context in which most people encounter it. The synthesis begins in the skin, where ultraviolet B radiation converts 7-dehydrocholesterol to pre-vitamin D3, which isomerizes thermally to vitamin D3. Vitamin D3, from synthesis or dietary intake, is biologically inert. It is hydroxylated in the liver to 25-hydroxyvitamin D3, calcidiol, which is the primary circulating storage form, and the form measured in blood tests, of vitamin D status. Calcidiol is then hydroxylated in the kidney by the enzyme CYP27B1 to 1,25-dihydroxyvitamin D3, calcitriol, the biologically active form. This final hydroxylation step is tightly regulated It is stimulated by parathyroid hormone and by low calcium or phosphate, and it is suppressed by FGF23, fibroblast growth factor 23, a phosphatinin hormone produced by bone, in response to vitamin D and phosphate loading that provides the negative feedback, limiting calcitriol production. Calcitriol acts through the nuclear vitamin D receptor, expressed in the intestinal epithelium, where it drives calcium and phosphate absorption, and in virtually every other tissue in the body, where it regulates cell proliferation, differentiation, and immune function. The breadth of vitamin D receptor expression, coupled with the high prevalence of vitamin D deficiency in populations living at high latitudes or with limited sun exposure, has fueled substantial research into the non-skeletal consequences of deficiency, with associations found between low vitamin D status and increased risk of autoimmune disease, certain cancers, cardiovascular disease, and infection. The enteroinsular axis, the communication between the gut and the pancreatic islets, mediated by the incretin hormones GLP-1 and GIP, produces the incretin effect, in which an oral glucose load stimulates much more insulin secretion than the same glucose administered intravenously, because oral glucose triggers incretin release from the gut that potentiates the pancreatic beta cell's response. The incretin effect was first noticed as an empirical observation. Glucose given by mouth provoked a much larger insulin response than the same amount of glucose infused directly into the blood to produce the same plasma glucose concentration. The oral route produced more insulin than could be accounted for by the glucose signal alone. The explanation turned out to be the incretin hormones, gut peptides released in response to food in the intestinal lumen that signal the pancreatic islets to prepare for the incoming glucose load, enhancing their insulin secretory response. Two incretin hormones account for most of the incretin effect: GIP, glucose-dependent insulinotropic polypeptide, produced by K cells in the duodenum and jejunum, and GLP-1, glucagon-like peptide 1, produced by L cells in the ileum and colon. Both act on G-protein-coupled receptors on the beta cell to increase intracellular cyclic AMP and potentiate the glucose-dependent insulin secretion. Their action is crucially glucose-dependent. They amplify the insulin response to hyperglycemia, but do not stimulate insulin secretion when blood glucose is normal, which means that therapeutic exploitation of the incretin system does not cause hypoglycemia in the way that some insulin secretagogues do. The incretin effect accounts for approximately 50 to 70% of the total insulin secretion after an oral glucose load, and it is substantially reduced in type 2 diabetes, partly because the GLP-1 response is impaired, and partly because the beta cell's response to GLP-1 is blunted. The therapeutic restoration of GLP-1 activity through GLP-1 receptor agonists has become one of the most transformative developments in diabetes pharmacology. The adrenal medulla secretes adrenaline and noradrenaline in response to sympathetic nervous system activation, and these catecholamines act on adrenergic receptors throughout the body to produce the rapidly coordinated fight or flight response, increasing heart rate, redirecting blood to muscles, dilating the airways, and mobilizing glucose within seconds of a perceived threat. The fight or flight response is the body's emergency operating mode, the state of maximal physiological readiness for intense physical action that evolved to deal with the immediate threat of predators and conspecific violence, and that the modern nervous system continues to activate in response to psychological threats of all kinds, including the daily stressors of contemporary life that require no running and no fighting whatsoever. The adrenaline surge is mediated through multiple adrenergic receptor subtypes with different tissue distributions and different downstream effects. Beta-1 adrenergic receptors in the heart mediate the increase in heart rate and myocardial contractility, the racing heart of fear. Beta-2 receptors in the bronchial smooth muscle mediate airway dilation, the easier breathing of the aroused state, and the basis for beta-agonist bronchodilators used in asthma. Beta-2 receptors in skeletal muscle vasculature mediate vasodilation, redirecting blood from the central circulation to the working muscles. Alpha-1 receptors in the skin and gut vasculature mediate vasoconstriction, the pallor of fear, and the disruption of digestion. Beta-3 receptors in adipose tissue mediate lipolysis, the mobilization of fat stores for fuel. Alpha-2 receptors in the pancreatic beta cell inhibit insulin secretion transiently, because during the fight or flight response, insulin's glucose storage function is irrelevant. The priority is glucose mobilization. The overall effect of this multi-receptor orchestra is a body primed for action, ready to run or fight, with every physiological system oriented toward the maximal performance of intense physical exertion. The thyroid gland's unique ability to concentrate iodide from the blood against a concentration gradient of 25 to 1 or more is accomplished by the sodium iodide symporter, a membrane protein that uses the sodium gradient maintained by the sodium-potassium ATPase to drive the active transport of iodide into the follicular cell, and this concentrating mechanism is exploited clinically in the use of radioactive iodine to image and ablate thyroid tissue. The sodium iodide symporter is one of the most remarkable active transport mechanisms in the body, and its thyroid-specific expression has given medicine one of its most targeted therapeutic tools. The symporter co-transports 2 sodium ions and 1 iodide ion into the follicular cell, using the energy of the inward sodium gradient maintained by the NAK-ATPase to drive the uphill transport of iodide against its electrochemical gradient. The resulting intracellular iodide concentration, 25 to 40 times the plasma concentration under basal conditions, and up to 200 times during TSH stimulation, provides the substrate for thyroid hormone synthesis. the thyroid-specific expression of the NIS importer means that radioactive isotopes of iodine, particularly iodine-131, are selectively taken up by thyroid tissue and by thyroid-derived cells. This selectivity is the basis for the clinical use of radioactive iodine. The oral administration of I-131 is taken up almost exclusively by thyroid tissue, where its beta emission destroys the follicular cells from within. This provides a highly targeted treatment for hyperthyroidism, ablating the overactive thyroid with minimal systemic radiation exposure, and for differentiated thyroid cancer, where metastatic disease in lymph nodes or distant sites can be treated with radioiodine as long as the cancer cells retain sufficient NIS expression. Thyroid imaging with I-123, which emits gamma radiation, suitable for external detection rather than beta particles, similarly exploits the NIS to produce images that reflect the distribution and activity of thyroid tissue throughout the body. The same molecular pump that feeds the thyroid gland with its iodine precursor has become, in clinical hands, both a therapeutic target and a diagnostic window. The gonads, the testes in males, and the ovaries in females respond to LH and FSH through a system that ensures the coordinated production of both the sex steroids and the gametes, with the spermatogonia and follicular cells using FSH for gamete development, while the Leydig cells and theca cells use LH for steroid synthesis. The gonad is a dual-function organ. It must simultaneously produce germ cells and produce sex steroids, and the coordination of these two functions requires a precisely organized cellular architecture and hormonal response system. In the testis, this duality is reflected in the two distinct cell populations. The Leydig cells, scattered in the interstitial tissue between the seminiferous tubules, respond to LH with testosterone production. The Sertoli cells, lining the seminiferous tubules, respond to FSH and to the local testosterone provided by the Leydig cells to support spermatogenesis. The Sertoli cells are the nurses of the developing sperm. They form the blood-testis barrier that creates the immune-privileged environment required for the genetically distinct sperm. They produce the androgen-binding protein that concentrates testosterone in the lumen of the tubule, and they produce inhibin B, a peptide that feeds back to the pituitary to suppress FSH secretion, providing the gonad-specific regulation of gonadotropin levels. In the ovary, the parallel dual system involves the theca cells of the follicular wall, which respond to LH with androgen production, and the granulosa cells, which respond to FSH with aromatase expression, converting the cal androgens to estradiol. This two-cell, two-gonadotropin model of ovarian estrogen synthesis requires the coordinated response of two adjacent cell types to two different pituitary signals, and its disruption by the inappropriate LH to FSH ratio of polycystic ovary syndrome produces the excess androgen relative to estrogen that characterizes that condition. The stress response to exercise, The physiological changes in hormone concentrations during and after physical activity involves a complex, coordinated hormonal program that includes rises in cortisol, growth hormone, catecholamines, and glucagon, and falls in insulin, together mobilizing the fuel substrates needed to power sustained physical effort. Exercise is, from the endocrine system's perspective, a controlled form of the stress response, a physiological challenge that requires fuel mobilization, cardiovascular adjustment, and eventually recovery and adaptation, all orchestrated through hormonal signals. Within seconds of the onset of intense exercise, the sympathoadrenal system activates adrenaline and noradrenaline from the adrenal medulla and sympathetic nerve terminals respectively, drive the cardiovascular adjustments, stimulate hepatic glucose output and glycogenolysis in muscle, inhibit insulin secretion, and stimulate glucagon release. The rising glucagon and falling insulin reverse the fed state metabolic program, mobilizing glucose from glycogen stores and fatty acids from adipose tissue for the fuel needs of working muscle. Growth hormone rises in proportion to exercise intensity, promoting fatty acid mobilization and opposing insulin's glucose storage action, conserving glucose for the brain during prolonged effort. Cortisol rises more slowly, peaking 30 to 60 minutes into prolonged exercise, and its sustained elevation supports gluconeogenesis, the synthesis of new glucose from amino acid precursors, providing a continuous glucose supply when glycogen stores are running low. The hormonal response to exercise is finely calibrated to intensity and duration. Brief intense exercise produces a sharp catecholamine surge and rapid fuel mobilization. Prolonged moderate exercise produces a more gradual cortisol rise and increasing fat mobilization as glycogen stores are progressively depleted. The recovery phase following exercise involves its own hormonal program. Growth hormone remains elevated to promote protein synthesis and tissue repair. The sensitivity to insulin rises, one of the most important health effects of regular exercise, as the glucose transporters are upregulated in the skeletal muscle by the exercise bout. The negative feedback regulation of the HPA axis operates at multiple levels. Cortisol suppresses CRH in the hypothalamus, ACTH secretion from the pituitary, and adrenal responsiveness to ACTH, and the breakdown of this multilevel feedback in conditions of chronic stress by glucocorticoid receptor downregulation is a central feature of the HPA dysregulation associated with depression and burnout. The multilevel negative feedback of the HPA axis is designed for redundancy and precision, each level of the axis providing an additional layer of cortisol sensitivity to ensure that the stress response is appropriately terminated when it is no longer needed. The fast feedback occurs within minutes. Cortisol's membrane-acting effects on the pituitary corticotrophs suppress ACTH release rapidly. The intermediate feedback occurs over hours. Cortisol enters the hypothalamic neurons and downregulates CRH gene expression, reducing the hypothalamic drive to the pituitary. The slow feedback, operating over days, involves the downregulation of glucocorticoid receptor expression in the hippocampus, hypothalamus, and pituitary, reducing the sensitivity of the feedback system itself. This slow downregulation is the molecular mechanism by which chronic stress dysregulates the HPA axis. Extended cortisol elevation reduces the sensitivity of the feedback receptors, making the axis less responsive to cortisol's suppressive signal and allowing cortisol levels to remain inappropriately elevated. The hippocampus, which expresses high levels of glucocorticoid receptors and exerts an inhibitory tone on the HPA axis through projections to the hypothalamus, is particularly vulnerable to chronic glucocorticoid exposure, which reduces glucocorticoid receptor expression and, over time, damages hippocampal neurons. The reduced hippocampal volume, found in patients with major depression and with post-traumatic stress disorder, consistently observed across many imaging studies, may partly reflect this glucocorticoid-mediated hippocampal damage, providing a structural substrate for the memory and cognitive problems associated with chronic stress states. The thymus, the gland that produces T lymphocytes during childhood and adolescence, is itself an endocrine organ, producing thymosin and thymopoietin peptides that regulate T-cell development and maturation, and that continue to have immunomodulatory effects, even after the thymus has largely involuted in adulthood. The thymus occupies a dual role that is not always appreciated. In either immunological or endocrine contexts, it is simultaneously the primary site of T lymphocyte development, the school in which T cells learn to distinguish self from non-self, and an endocrine gland, whose hormonal products coordinate this educational process and extend their effects into the periphery. The thymus is most active in childhood, reaching its greatest size relative to body weight in the first years of life, and it begins to involute, to be replaced by fat tissue, after puberty, with the involution driven in part by the rising sex steroids of pubescence. The thymic epithelial cells produce a family of peptide hormones, thymosin alpha-1, thymosin beta-4, thymulin, thymopoietin, that regulate the maturation, proliferation, and selection of T-cell precursors within the thymic cortex and medulla, and that are secreted into the bloodstream, where they can influence T-cell function in the periphery. Thymulin, a 9-amino acid peptide that requires zinc for its biological activity, stimulates the differentiation of T-cell precursors, promotes T-cell proliferation in response to antigenic stimulation, and enhances natural killer cell activity. The age-related decline in thymulin levels, paralleling the involution of the thymic tissue that produces it, has been proposed as a contributor to the immunosenescence of aging, the gradual decline in immune function that increases the susceptibility to infection and cancer, and reduces the effectiveness of vaccination in elderly individuals. The recognition of the thymus as an endocrine organ adds another dimension to its role as the immune system's university, It does not merely educate T cells in resonance, but sends hormonal signals into the body to continue that education at a distance. The discovery that the heart is an endocrine organ, producing atrial natriuretic peptide in response to atrial stretch, caused by elevated blood volume, expanded the concept of the endocrine system beyond its classical glandular definition, and was one of the first demonstrations that virtually any organ can acquire hormonal function when the need arises. The heart as a pump is the image of cardiac function that dominated medicine for centuries. The heart as a gland, producing and secreting hormones, was a concept that did not enter the mainstream until 1981, when Adolfo de Bold and colleagues demonstrated that injection of atrial extract into rats produced a rapid, powerful natriuresis, a dramatic increase in sodium excretion in the urine, and hypotension. The active principle turned out to be a peptide, now called atrial natriuretic peptide, produced by the cardiomyocytes of the atria and stored in secretory granules visible on electron microscopy, unmistakable evidence of an endocrine cell in what had been regarded as purely mechanical tissue. ANP is released when the atrial walls are stretched by elevated blood volume or elevated atrial filling pressure, the cardiomyocytes acting as mechanosensors that detect the volume overload and respond hormonally. ANP acts on the kidney to increase sodium and water excretion, on the adrenal to suppress aldosterone secretion, and directly on blood vessels to cause vasodilation, together reducing blood volume and blood pressure, opposing the fluid retention signals of the renin-angiotensin-aldosterone system. A related peptide, brain natriuretic peptide, despite its misleading name, produced primarily by the ventricular myocardium in response to pressure overload and ventricular stretch, has become one of the most useful clinical biomarkers. In heart failure, its blood levels rising in proportion to the degree of cardiac stress, and providing the cardiologist with a liquid biopsy of the heart's mechanical burden. The discovery of the heart as an endocrine organ set a precedent that has since been extended to the muscle, the bone, the liver, and many other tissues. The gut-brain axis, the bidirectional communication between the gastrointestinal tract and the central nervous system through hormonal, neural, and immune pathways, is increasingly recognized as a fundamental system governing not only digestive function, but also mood, cognition, appetite, and the biological basis of the gut feelings that are so central to human experience. The gut-brain axis is not simply the vagal afferent pathway or the enteric nervous system. It is a multimodal communication network operating through at least 3 parallel channels. The neural channel involves the vagus nerve, whose afferent fibers carry information from the gut's mechanoreceptors, chemoreceptors, and enteroendocrine cells to the brainstem nucleus tractus solitarius, which distributes this information to the hypothalamus, amygdala, cortex, and the brainstem autonomic centers. The hormonal channel involves the gut hormones, GLP-1, CCK, ghrelin, PYY, circulating in the blood to act on the hypothalamus and other brain regions. And an immune channel involves the gut microbiome's metabolic products, short-chain fatty acids, tryptophan metabolites, lipopolysaccharide, crossing the gut epithelium and acting locally on immune cells and enteroendocrine cells, and reaching the systemic circulation in the brain. The gut's enteroendocrine cells, the most numerous endocrine cells in the body, scattered through the epithelial lining from stomach to colon, are exquisitely sensitive to the luminal contents, detecting specific nutrients, microbial metabolites, bile acids, and pH, and responding with precisely calibrated hormonal releases that signal the brain, the pancreas, and the rest of the digestive tract about the current contents and nutritional status of the gut. The gut feelings of everyday language, the butterflies of anxiety, the nausea of disgust, the hollow feeling of grief, the warmth of satiety, all have their partial basis in this hormonal and neural dialogue between the gut and the brain. The gut is not simply where food is processed. It is where the body reads what has been consumed and communicates that reading to the mind. The concept of hormonal crosstalk, the ability of hormones from one system to influence the sensitivity, production, or action of hormones from another system, reveals the endocrine system not as a collection of independent axes, but as an integrated network in which every hormonal signal exists in the context of all the others. Hormonal crosstalk is so pervasive that it is almost easier to list the hormonal systems that are truly independent and to find none than to enumerate the specific interactions. A few prominent examples illustrate the principle. Thyroid hormones increase the expression of beta-adrenergic receptors in the heart, which is why hyperthyroidism produces tachycardia, and why patients with hypothyroidism tolerate cardiovascular stress poorly. Cortisol increases the expression of glucagon receptors in the liver, which is why the insulin resistance of glucocorticoid excess is not solely a direct glucocorticoid effect, but also involves potentiating glucagon's glucose-raising action. Estrogen increases the synthesis of thyroid-binding globulin, which is why women on oral contraceptives, or estrogen therapy, often have elevated total thyroid hormone levels without being hyperthyroid, because the increased protein binding reduces the free hormone fraction and triggers compensatory TSH-driven increases in production. Insulin inhibits GnRH pulsatility, which is part of the mechanism by which hyperinsulinemic states like polycystic ovary syndrome disrupt the normal reproductive hormonal pattern. Leptin is permissive for the activity of the hypothalamic pituitary gonadal axis. Falling leptin in states of energy deficiency, such as starvation or excessive athletic training, suppresses GnRH pulsatility and leads to amenorrhea, which is part of the functional hypothalamic suppression of reproduction when energy stores are insufficient to sustain a pregnancy. The endocrine system is, in this sense, a distributed decision system, integrating information from dozens of hormones simultaneously, to produce an integrated assessment of the body's state and to generate the coordinated hormonal response that the current state requires. The concept of pulsatile hormone secretion, the release of many hormones in discrete pulses, rather than at a constant rate, is not merely a quirk of secretory biology, but a functional necessity, because continuous exposure of receptors to their ligand produces receptor downregulation and desensitization, while pulsatile exposure maintains receptor sensitivity. Pulsatile secretion is the default pattern of most hypothalamic-releasing hormones and many pituitary hormones, and the functional importance of this pattern was elegantly demonstrated in the context of GnRH therapy for reproductive medicine. GnRH, the hypothalamic decapeptide that drives the pituitary to release LH and FSH, is normally secreted in pulses, approximately once per hour, and the pituitary gonadotrophs are exquisitely sensitive to this pulsatile input. When GnRH was first used therapeutically to stimulate ovulation in women with hypothalamic amenorrhea, it was administered in pulsatile fashion by a portable pump delivering a pulse every 90 minutes, mimicking the natural pattern. This produced normal gonadotropin secretion and successful ovulation. When the same total dose was administered as a continuous infusion, the response was paradoxical. LH and FSH secretion was suppressed. The continuous GnRH caused the GnRH receptors on the gonadotrophs to be downregulated and desensitized. After the initial stimulation, the pituitary became unresponsive. This insight turned out to have an unexpected therapeutic application: the development of long-acting GnRH agonists that, given as monthly depot injections, produce continuous, non-pulsatile GnRH receptor stimulation, and therefore paradoxical suppression of the gonadal axis. These agents are now widely used to suppress sex steroid production in prostate cancer, in precocious puberty, and in gender-affirming hormone therapy. The pulse, in hormonal biology, is not incidental. It is essential. The rhythm carries as much information as the signal itself. The measurement of hormone levels in the blood is complicated by the fact that most steroid hormones and thyroid hormones circulate predominantly bound to carrier proteins, with only the tiny free fraction being biologically active, and conditions that alter the concentration of these carrier proteins can produce dramatic changes in total hormone levels without any change in the biologically active free fraction. The concept of hormone binding to plasma proteins is one of the more clinically treacherous aspects of endocrine physiology, because total hormone measurements, the most commonly performed clinical assays, can be misleading in patients whose carrier protein concentrations are abnormal. Sex hormone binding globulin, SHBG, transports both testosterone and estradiol with high affinity, and its concentration in the blood is regulated by many factors. It is increased by estrogen, thyroid hormones, and certain liver conditions. It is decreased by androgens, insulin, and obesity. A woman taking oral estrogen will have elevated SHBG, elevated total testosterone and estradiol, but normal free testosterone and estradiol, because the increased binding protein is sequestering more of the circulating steroid. An obese man will have reduced SHBG, reduced total testosterone, but the free testosterone may be relatively preserved. Cortisol-binding globulin, CBG, transports cortisol, and its concentration, also estrogen-sensitive, can affect interpretation of cortisol measurements. Thyroid-binding globulin transports the thyroid hormones, and its increase in pregnancy, driven by the estrogen of gestation, produces the elevated total T4 seen in normal pregnancy. That would suggest hyperthyroidism, if not recognized as a protein-binding artifact. The clinical solution is to measure free hormone concentrations directly, free T4, free testosterone, but these assays are technically challenging and expensive. More commonly, the clinical interpretation of hormone levels must account for the likely carrier protein status of the patient, adding a layer of biochemical reasoning to what might otherwise seem like a simple laboratory result. The concept of hormone resistance, in which the target tissues fail to respond normally to a hormone despite its presence in normal or elevated concentrations, represents a fundamentally different category of endocrine disorder from hormone deficiency, and its discovery in the context of insulin resistance, thyroid hormone resistance, and androgen insensitivity has illuminated the receptor and post-receptor mechanisms of hormone action. Hormone resistance was a concept that took time to establish, precisely because it seems to violate a simple expectation: if a hormone is present, it should work. The discovery that it sometimes does not, that tissues can become unresponsive to hormones, despite their normal or elevated presence, required the conceptual separation of the hormone itself from its receptor and from the post-receptor signaling machinery that translates receptor activation into biological effect. The most clinically prevalent form is insulin resistance, in which the post-receptor signaling pathway is impaired through multiple mechanisms, including inflammatory cytokine signaling, lipid-induced inhibition of the IRS proteins, and mitochondrial dysfunction. The result is that normal insulin concentrations cannot produce normal glucose uptake, requiring compensatory hyperinsulinemia to maintain glucose homeostasis. Thyroid hormone resistance, caused by mutations in the thyroid hormone receptor beta gene, produces a state in which tissues are variably resistant to thyroid hormone action, with the most dramatic phenotype in the pituitary, which fails to be suppressed normally by circulating T3 and continues to drive high TSH levels. The elevated TSH drives thyroid hormone overproduction, producing elevated T3 and T4 alongside elevated TSH, a biochemical combination that would normally be impossible, and that confused clinicians for years before the molecular basis was understood. Androgen insensitivity syndrome, caused by mutations in the androgen receptor gene, produces individuals with 46 XY chromosomes and fully functional testes that produce normal testosterone. But because the androgen receptor cannot respond, the testosterone has no effect, and the phenotype is completely female. The hormone is present. The receptor is absent. The effect is zero. Hormone resistance reveals, more clearly than any other class of endocrine disorder, that the hormone is only the beginning of the story. The concept of autocrine and paracrine signaling, in which hormones act on the cells that produce them, or on immediately adjacent cells, rather than on distant targets via the bloodstream, extends the definition of hormonal action beyond the classical endocrine model and reveals that many tissues govern their own function through local chemical conversations. The classical endocrine model: gland secretes hormone into the blood, blood carries hormone to distant target organ, is the most familiar mode of hormonal signaling, but it is by no means the only one. Paracrine signaling, in which a secreted molecule acts on adjacent cells within the same tissue, is as fundamental to tissue function as the long-range endocrine signal. The paracrine interactions within the pancreatic islets are a well-studied example. The insulin produced by the beta cells acts on adjacent alpha cells to suppress glucagon release, a local paracrine loop that contributes to the coordinate regulation of the two hormones. The somatostatin produced by the delta cells of the islets acts paracrinely on both the alpha and beta cells to suppress their hormone secretion, another local regulatory loop. Autocrine signaling, in which a cell's secreted molecule acts back on the same cell through its surface receptors, is important in development and in cancer biology. Growth factors that are both produced and responded to by the same cell can drive autonomous proliferation, a feature of many cancers. In reproductive endocrinology, the paracrine signaling between the granulosa cells and the oocyte within the ovarian follicle, the oocyte producing factors that regulate granulosa cell differentiation, the granulosa cells producing factors that regulate oocyte maturation, is essential for normal follicle development and oocyte quality. These local conversations within the tissue sit alongside the systemic hormonal signals as parallel layers of regulation, allowing tissues to self-regulate their local environment while remaining responsive to the body's broader hormonal context. The hormone receptor interaction is governed by two key parameters: affinity, the tightness with which the hormone binds the receptor, and receptor number, the density of receptors on the target cell, both of which are subject to regulation and determine the sensitivity of the target cell to the hormonal signal, independently of the hormone's circulating concentration. The cellular response to a hormone depends not only on the hormone's concentration in the blood, but on the sensitivity of the target cell, and sensitivity is governed by the affinity and the number of the receptors available. Both parameters are regulated by physiological and pathological processes, allowing the body to adjust the responsiveness of its tissues to hormonal signals without changing the signal itself. Receptor downregulation, The reduction in receptor number, in response to chronic exposure to a high ligand concentration, is a general phenomenon that protects cells from overstimulation. Insulin receptors are downregulated in hyperinsulinemia, contributing to insulin resistance. Adrenergic receptors are downregulated with chronic sympathetic activation, which is why beta blocker therapy, when discontinued abruptly, can produce rebound hypertension and tachycardia, as the newly exposed, previously downregulated receptors respond to normal levels of catecholamines with excessive sensitivity. Thyroid hormone receptors are upregulated in hypothyroidism, the cell increasing its receptor density to maximize the response to the limited available hormone. Receptor affinity, the binding constant that determines how effectively a given hormone concentration occupies the receptor, can also be regulated by post-translational modifications of the receptor itself, by the lipid composition of the membrane in which the receptor is embedded, and by receptor dimerization, oligomerization, or interaction with allosteric modulators. The target cell is not passive in the face of hormonal signals. It is continuously adjusting its sensitivity to maintain appropriate responsiveness across a range of ligand concentrations, and these adjustments are themselves part of the homeostatic machinery of the endocrine system. The phenomenon of chronobiology, the study of biological timing in circadian rhythms, intersects profoundly with endocrinology, because virtually every major hormonal axis shows a circadian pattern of secretion that reflects the biological clock's anticipation of the metabolic, reproductive, and stress-related demands of the 24-hour day. The temporal architecture of the endocrine system is as fundamental to its function as the nature of its constituent hormones. Virtually every hormone measured in the blood shows significant variation across the 24-hour day, not merely the well-known patterns of cortisol and melatonin, but testosterone, GH, TSH, leptin, ghrelin, insulin secretion, prolactin, and many others. These circadian hormonal rhythms are generated partly by the suprachiasmatic nucleus clock, which communicates to peripheral endocrine glands through both direct neural pathways and hormonal intermediaries, and partly by the peripheral circadian clocks running in the endocrine glands themselves. The temporal coordination of the hormonal system means that the same hormone can have different effects depending on the time of day. Cortisol's anti-inflammatory effect is stronger at some phases of the clock than others. It also means that the metabolic response to the same meal is different at different times of day. A calorie consumed in the morning is handled differently than the same calorie consumed in the evening, partly because the insulin secretory response, the incretin effect, and the sensitivity of peripheral tissues to insulin all vary with the circadian phase. This is the biological basis for the growing interest in chrononutrition, the study of how the timing of food consumption interacts with the circadian system to affect metabolic outcomes. And it is the reason why shift work in circadian misalignment, exposure to light at the wrong times, eating at the wrong phases of the clock, produce metabolic dysregulation. The hormonal system is being asked to operate in a temporal environment misaligned with its internal timing. Tonight, your hormonal system is in alignment. The melatonin is rising. The cortisol is low. The growth hormone pulse is preparing. The body knows what time it is. FGF21, fibroblast growth factor 21, is a metabolic hormone produced primarily by the liver in response to fasting, ketogenic diet, and exercise that acts on fat tissue in the brain to promote fat mobilization, increase energy expenditure, suppress appetite for simple carbohydrates, and induce a metabolic state adapted to low carbohydrate availability. FGF21 is a member of the fibroblast growth factor family, a large group of signaling proteins with diverse biological functions, but its role in metabolism distinguishes it from its relatives. It is produced primarily by hepatocytes in response to the transcriptional activation of its gene by PAR, peroxisome proliferator activated receptor alpha, which is activated during fasting, and by fatty acid accumulation in the liver, making FGF21 essentially a fasting signal. Acting on the adipose tissue through the coreceptor beta-klotho, FGF21 promotes lipolysis and fatty acid release, providing the fuel for the ketogenesis that the fasting liver conducts. Acting on the brain, particularly the hypothalamus and the cortex, FGF21 appears to suppress the preference for sweet foods and alcohol, a somewhat surprising behavioral effect that has attracted substantial pharmaceutical interest. The reciprocal relationship between carbohydrate intake and FGF21. Carbohydrate suppresses FGF21, while carbohydrate restriction and fasting induce it, appears to be one of the mechanisms by which very low carbohydrate diets reduce the appetite for carbohydrates over time. The FGF21 induced by the dietary restriction is itself changing the brain's hedonic response to sweet foods. FGF21 also has beneficial effects on insulin sensitivity and lipid metabolism, making it a drug target of considerable interest in metabolic disease. The liver, in producing FGF21 during fasting, is signaling to the rest of the body that the carbohydrate supply has been cut and that metabolic adaptation is required, a fasting hormone that prepares the system for the switch to fat-based fuel. The concept of endocrine disruptors, synthetic chemicals in the environment that interfere with the normal function of the endocrine system by mimicking hormones, blocking receptors, or altering hormone synthesis and metabolism, has emerged as a significant concern in public health, with evidence linking certain exposures to disrupted reproductive development, metabolic disease, thyroid dysfunction, and cancer. Endocrine disruptors are chemicals that interact with the endocrine system in unintended ways, either by resembling naturally occurring hormones well enough to activate hormonal receptors, by blocking hormone receptors and preventing the natural hormone from acting, by altering the synthesis or metabolism of endogenous hormones, or by modifying the expression of hormone receptors. The list of chemicals with endocrine disrupting properties includes industrial chemicals, bisphenol A in plastics, phthalates in flexible plastics, polychlorinated biphenyls, pesticides and herbicides, pharmaceuticals excreted into wastewater, and components of personal care products. The endocrine system's vulnerabilities to these compounds reflect its dependence on precise molecular recognition. The hormone receptor interaction requires specific structural complementarity, but this complementarity can be approximated by structurally similar synthetic chemicals that were not designed with receptors in mind. The concern about endocrine disruptors is particularly acute in the context of developmental exposures. The endocrine system plays essential roles in fetal and neonatal development, including the sexual differentiation of the brain and genitalia, the programming of the metabolic set points, and the development of the immune system, and disruption of hormonal signals during these critical windows can have permanent consequences that are not apparent until later in life. The epigenetic programming effects of early endocrine disruption may even persist into the next generation through germline transmission. This complexity, the long latency between exposure and effect, the population-level statistical signal amid individual variation, and the ubiquity of many exposures makes the study of endocrine disruptors scientifically and regulatorily challenging. There are so many places left to go in the endocrine system: the chemistry of growth, the hormones of sleep, the signals of bone, the remarkable endocrine functions of the heart, and the immune system, and the brain itself. If you would like to be here for those journeys, and for all the other quiet wonders that Relax with Science brings to the night, one slow evening at a time, subscribing keeps the door open between us. Something will be waiting on an evening when you need it. Made slowly, made carefully, made for exactly this hour. The neuroendocrine tumors, cancers arising from hormone-producing cells, scattered throughout the body in the diffuse neuroendocrine system, demonstrate that endocrine function is distributed far more widely than the classical glands, with tumors arising in the pancreas, the gut, the lung, the thyroid, the adrenal, and many other sites, producing excess hormones whose systemic effects can be dramatic and diagnostically challenging. The diffuse neuroendocrine system, the population of hormone-producing cells, distributed throughout the body outside the classical endocrine glands, is as clinically important as the glands themselves, and its tumors provide some of the most fascinating and challenging cases in endocrinology. The carcinoid tumor, arising from enterochromaffin cells in the gut that normally produce serotonin, can cause the carcinoid syndrome when it metastasizes to the liver, flushing, diarrhea, and bronchoconstriction produced by the serotonin and other vasoactive amines that, once the liver can no longer metabolize them, reach the systemic circulation. Insulinomas, tumors of the pancreatic beta cells that autonomously secrete insulin regardless of blood glucose, produce recurrent episodes of hypoglycemia, typically in the fasting state, whose dramatic neuroglycopenic symptoms, confusion, sweating, loss of consciousness, can be mistaken for neurological or psychiatric disease before the hormonal cause is identified. Glucagonomas, alpha cell tumors, produce a syndrome of diabetes, weight loss, and a characteristic migratory rash called necrolytic migratory erythema. Gastrinomas, G-cell tumors usually arising in the duodenum or pancreas, produce excess gastrin, which drives the acid hypersecretion of the Zollinger-Ellison syndrome. The study of these tumors has illuminated normal endocrine physiology, observing what happens when a particular hormone is produced in excess without regulation, provides a natural experiment whose results clarify the hormone's normal physiological roles. The neuroendocrine tumor is, in this sense, a pathological window into the endocrine system's normal functions. The hormonal regulation of puberty, initiated by the pulsatile reactivation of the hypothalamic GnRH pulse generator after the quiescence of childhood, requires the coordinated release of kisspeptin from hypothalamic neurons that act directly on the GnRH neurons, and mutations in the kisspeptin GPR54 system produce hypogonadotropic hypogonadism with absent puberty. The hypothalamic GnRH pulse generator is active in the fetus and in the first months of life, the brief activation known as the mini-puberty of infancy, and then falls quiescent during childhood, suppressed by neural mechanisms that are not fully understood, but that include the active inhibitory activity of GABergic neurons and the restraint of kisspeptin neurons. The reactivation of the pulse generator at the onset of puberty, producing the rising LH pulses, initially nocturnal and episodic, that characterize early puberty, requires the removal of this inhibition and the progressive activation of the kisspeptin neurons. Kisspeptin is a 54 amino acid peptide produced by two populations of hypothalamic neurons, the arcuate nucleus kisspeptin neurons, which also produce neurokinin B and dynorphin, the KND neurons, and which generate the GnRH pulse through rhythmic self-stimulation and self-inhibition, and the anteroventral periventricular kisspeptin neurons, which are responsible for the estrogen-positive feedback and the LH surge. The critical importance of the kisspeptin GPR54 system, the receptor through which kisspeptin signals to the GnRH neurons, was revealed by loss-of-function mutations in both kisspeptin and its receptor, which produce hypogonadotropic hypogonadism, absent puberty, absent sex steroids, and infertility, despite normal GnRH neurons that simply receive no activating signal. The discovery of kisspeptin as the master regulator of GnRH pulsatility, and therefore of puberty, reproductive cycling, and the seasonal regulation of reproduction in species that breed seasonally, has been one of the major advances in reproductive neuroendocrinology. The concept of the endocrine clock, the idea that the endocrine system, participates in the biological timing of the aging process, Through the age-related decline of GH and IGF-1, DHEA, sex steroids, and other hormones, has driven both the scientific investigation of the hormonal basis of aging and the commercial exploitation of hormone replacement as an anti-aging strategy. The hormonal landscape of the body changes dramatically across the lifespan in a pattern that is reproducible enough to serve as a biomarker of biological age. Growth hormone and IGF-1 levels, high in childhood and adolescence, begin to decline in early adulthood and continue to fall throughout life, a process called somatopause. DHEA and its sulfate, produced by the adrenal zona reticularis, peak in the mid-20s and decline progressively, reaching less than 20% of their peak values by the 8th decade, in a process called adrenopause. Sex steroids fall dramatically at menopause in women, and more gradually in men. The andropause of testosterone decline, and in both sexes with advancing age. Melatonin production declines with age, contributing to the circadian disruption and sleep changes of aging. The question of whether these hormonal declines are causes of the aging phenotype, reduced muscle mass, increased body fat, reduced bone density, cognitive decline, reduced vitality, or consequences of the same aging processes that produce the clinical features, is central to the science of hormonal aging. The partial restoration of GH and IGF-1 levels through GH injections in GH-deficient older adults does improve body composition and reduces fat mass, but the evidence that it slows the fundamental aging process is lacking. The temptation to see the age-related hormonal declines as correctable deficiencies rather than adaptive changes of normal aging has driven a substantial anti-aging industry whose scientific basis remains contested. The interaction between the immune system and the endocrine system, neuroendocrine immunology, reveals a bidirectional communication in which hormones regulate immune function and immune signals regulate hormone secretion, with glucocorticoids as the most clinically important immunomodulatory hormones, and with pro-inflammatory cytokines as potent activators of the HPA axis. The endocrine system and the immune system are not separate physiological domains, but deeply interpenetrating systems that continuously modulate each other's activity. The glucocorticoids, primarily cortisol, are the endocrine system's most powerful immunological regulators, with effects that include the suppression of virtually every component of the innate and adaptive immune response, reduced cytokine production by macrophages and lymphocytes, reduced migration of neutrophils and macrophages to sites of inflammation, reduced antibody production by B cells, reduced T-cell proliferation, and increased apoptosis of immune cells. These effects are why synthetic glucocorticoids are among the most potent anti-inflammatory and immunosuppressive drugs available, and why their pharmacological doses, sustained over time, produce the infections and immune deficiencies of steroid therapy. In the opposite direction, the pro-inflammatory cytokines, particularly IL-1, IL-6, and TNF-alpha, are potent activators of the HPA axis, directly stimulating CRH release from the hypothalamus and ACTH release from the pituitary, increasing cortisol levels in the context of infection or inflammation. This cytokine-driven HPA activation is part of the acute phase response to infection. The rising cortisol is both a metabolic response to the energy demands of the immune response and a regulatory feedback that prevents the immune activation from becoming uncontrolled. The sex steroids also modulate immune function in ways that contribute to the well-documented sex differences in autoimmune disease. Estrogen tends to enhance adaptive immunity and increase susceptibility to autoimmune conditions, while testosterone is generally immunosuppressive. Fibroblast growth factor 23, FGF23, produced by osteocytes in bone in response to phosphate loading, and vitamin D, acts on the kidney to reduce phosphate reabsorption and to suppress the final hydroxylation step of vitamin D activation, providing the phosphate-vitamin D regulatory axis that governs mineral metabolism, alongside the PTH vitamin D axis. FGF23 is a relatively recently discovered hormone. Its identification in the late 1990s through the study of rare genetic disorders of phosphate metabolism that has substantially revised the understanding of mineral homeostasis by revealing a bone-derived hormone that governs kidney function. Before FGF23's discovery, the regulation of phosphate was understood primarily through the phosphaturic effects of PTH and through dietary considerations. The identification of FGF23 as a phosphatonin, a hormone that reduces serum phosphate by increasing urinary phosphate excretion, revealed that bone itself was an active endocrine organ participating in mineral homeostasis. FGF23 is produced by osteocytes and osteoblasts in response to high serum phosphate, high 1,25-dihydroxyvitamin D, and high dietary phosphate intake. Acting on the kidney through the FGF receptor and the coreceptor klotho, FGF23 reduces the expression of the sodium phosphate cotransporters responsible for phosphate reabsorption in the proximal tubule, increasing urinary phosphate excretion and lowering serum phosphate. It also suppresses the CYP27B1 enzyme, responsible for the final activation of vitamin D to calcitriol, reducing the intestinal calcium and phosphate absorption that would otherwise add to the mineral load. Mutations in FGF23 or klotho produce the phosphate-wasting disorders, X-linked hypophosphatemia, and tumor-induced osteomalacia, characterized by excessive renal phosphate loss, and the bone softening that results from phosphate deficiency, and the elevation of FGF23 in chronic kidney disease, as the failing kidney's reduced ability to excrete phosphate drives FGF23 production, has been recognized as a contributor to the cardiac complications of kidney disease. The incretin hormones GLP-1 and GIP have been pharmacologically exploited in the treatment of type 2 diabetes and obesity through GLP-1 receptor agonists and dual GLP-1/GIP receptor agonists, producing the most effective pharmacological treatments for weight loss currently available, and demonstrating that hormonal signaling pathways can be therapeutically amplified with transformative clinical results. The GLP-1 receptor agonists, liraglutide, semaglutide, exenatide, and related compounds, are among the most significant pharmaceutical developments in endocrinology of recent decades, and their story illustrates the path from the discovery of a physiological hormone to a transformative drug. GLP-1's insulinotropic effects, its suppression of glucagon, its slowing of gastric emptying, its appetite-suppressing action in the hypothalamus, and its satiety-inducing effects in the gut, all make it an ideal target for diabetes and obesity pharmacology. But native GLP-1 has a plasma half-life of approximately 2 minutes, rapidly degraded by the enzyme DPP-4, too short for therapeutic use. The pharmaceutical solution was to develop structural analogs resistant to DPP-4 degradation, exendin-4 from the Gila monster salivary gland, and then fully synthetic analogs with fatty acid attachments for albumin binding, extending the half-life to days. Once-weekly subcutaneous injection of semaglutide produces circulating GLP-1 receptor agonist levels far higher than natural GLP-1 ever achieves, producing greater satiety, greater appetite suppression, and greater weight loss than could be achieved by modestly amplifying the natural signal. The dual GLP-1/GIP receptor agonists, tirzepatide being the first approved, produce even greater weight loss by simultaneously activating both incretin receptors, with clinical trials demonstrating average weight loss of 20 to 25% of body weight. These results, approaching the weight loss seen with bariatric surgery, represent a pharmacological achievement that would have seemed implausible a decade ago, achieved through the hormonal pharmacology of the gut-brain axis. The endocrine pancreas, the islets of Langerhans, representing only approximately 1 to 2% of the total pancreatic mass, contains 5 distinct cell types, each producing a specific hormone, and the local paracrine interactions among them fine-tune the hormonal output of the islet in response to the nutritional and metabolic state of the moment. The islet of Langerhans is a miniature endocrine organ within the exocrine organ of the pancreas, a sphere of perhaps 1,000 to 2,000 cells, richly vascularized and densely innervated, exquisitely sensitive to the chemical environment of the portal blood that flows through and around it. The 5 cell types are distributed in a characteristic concentric pattern in rodents, and in a more complex, intermingled pattern in humans that reflects the paracrine relationships among them. The beta cells, producing insulin, form the largest population, approximately 60 to 80% in humans, and are distributed throughout the islet. The alpha cells, producing glucagon, form approximately 15 to 20%. The delta cells, producing somatostatin, constitute approximately 5%. The PP cells of the pancreatic polypeptide, concentrated in the head of the pancreas, and the epsilon cells producing ghrelin, present in small numbers, particularly in fetal and neonatal islets, complete the complement. The paracrine interactions are as important as the endocrine ones for islet function. Insulin suppresses glucagon release from adjacent alpha cells. Somatostatin from the delta cells suppresses both insulin and glucagon release. Glucagon stimulates insulin secretion from adjacent beta cells, contributing to the first phase insulin release in response to a mixed meal. These local conversations within the islet are part of the coordinated islet response to nutritional stimuli and their disruption by the loss of beta cells in type 1 diabetes or by the inflammation of pancreatitis alters not just the missing cell's hormone, but the output of all the cells that depended on its paracrine signals. I wonder how you are feeling right now, whether the chemistry of rest has been finding its way through the body since you arrived here. Whether the melatonin rising quietly in your blood has begun to have its effect. Whether the breath is a little slower, the thoughts a little softer, the weight of the day a little further away. If it has, even slightly, I am glad. And if you feel like leaving something in the comments about what tonight feels like for you, I would find that genuinely meaningful. Sometimes the most important thing is simply to say, I was here. The chemistry was kind. That is enough. The hormone osteocalcin, produced by osteoblasts and secreted into the bloodstream, has been identified as a bone-derived hormone that promotes insulin secretion, improves insulin sensitivity in muscle, supports male fertility through its effects on the testes, and enhances physical performance during exercise, revealing bone as an endocrine organ with unexpectedly broad metabolic influence. The discovery of osteocalcin as a bone-derived metabolic hormone, part of the work of Gerard Carsendy and his colleagues, over the past 2 decades, has extended the endocrine map of the body to include the skeleton as an active participant in metabolic regulation. Osteocalcin is synthesized by osteoblasts in a form that is carboxylate, vitamin K-dependent carboxylation of specific glutamate residues, producing the form that binds hydroxyapatite and becomes incorporated into the bone matrix. But a proportion of osteocalcin is also released in an undercarboxylated form that does not bind bone matrix and instead enters the bloodstream where it acts as a hormone. The undercarboxylated form, the active hormonal form, increases insulin secretion from pancreatic beta cells, increases the insulin sensitivity of muscle cells, and promotes their glucose uptake. It has effects on the testes. The Leydig cells express osteocalcin receptors, and osteocalcin promotes testosterone synthesis, connecting bone metabolism and male reproductive function in an unexpected hormonal loop. Most intriguing is the evidence that osteocalcin levels rise during exercise and that osteocalcin is required for the full exercise-induced increase in energy availability and performance in mice. Animals lacking osteocalcin show impaired exercise capacity, and the exercise incapacity can be rescued by osteocalcin administration. This suggests that the skeleton, physically stressed by exercise, signals back through osteocalcin to mobilize the metabolic resources needed for the exercise effort. Bone does not merely respond to the forces of movement. It participates in the body's metabolic response to those forces. The hormonal response to fasting, the physiological adaptation to the absence of food, involves a coordinated decline in insulin, rise in glucagon, rise in cortisol and growth hormone, and eventually a rise in ketone bodies that serve as the brain's alternative fuel, representing one of the most elegant examples of multi-hormone coordination in human physiology. Fasting is the ancestral human experience, the state of not having eaten for hours or days. That was the inevitable condition of hunter-gatherer life, and the body has developed a hormonal program for managing it. That is, in its coordination and elegance, a masterwork of endocrine systems thinking. The response unfolds in phases. In the first hours after a meal, as blood glucose falls from its postprandial peak, insulin falls and glucagon rises. The liver responds to the high glucagon to insulin ratio by activating glycogenolysis, breaking down the stored glycogen to glucose, and suppressing the glycolytic and glycogen synthetic pathways. Blood glucose is maintained for 4 to 6 hours by this hepatic glycogen mobilization. As glycogen stores are depleted, the cortisol and growth hormone that have been rising in the latter part of the fast become the dominant signals. Cortisol drives gluconeogenesis in the liver, using amino acids from muscle proteolysis and glycerol from fat breakdown as substrates. Growth hormone promotes fat mobilization while sparing protein. By 24 hours of fasting, fat mobilization is the dominant fuel supply for most tissues. The fatty acids from adipose lipolysis, providing the fuel for muscle, heart, and liver, sparing glucose for the brain. As fasting extends, the liver converts fatty acids to ketone bodies, beta-hydroxybutyrate and acetoacetate, which can cross the blood-brain barrier and provide an alternative fuel for the brain, reducing the brain's glucose requirement and thereby reducing the need for gluconeogenesis and the muscle protein wasting it requires. The hormonal program of fasting is the body's ancient intelligence about scarcity, a system refined over millions of years of intermittent food availability, operating tonight in every person who has gone to bed without eating since dinner. The hypothalamic pituitary thyroid axis maintains thyroid hormone levels within a narrow range through a negative feedback loop of extraordinary sensitivity. TSH levels change by as much as 5 to 10-fold in response to changes in free T4 that are within the normal range, making TSH one of the most sensitive endocrine tests available and the primary screening tool for thyroid dysfunction. The sensitivity of the TSH response to small changes in free T4 is the clinical cornerstone of thyroid medicine. Because the relationship between TSH and free T4 is logarithmic rather than linear, a 10-fold change in TSH corresponds to a much smaller change in free T4. The TSH is exquisitely sensitive to the thyroid hormone level. Even within the normal range of free T4, TSH varies by several fold. At the edges of the normal range, TSH changes that would be considered abnormal reflect free T4 values that are barely outside normal. This log-linear relationship means that TSH is a much more sensitive indicator of the thyroid hormone status of the pituitary, and by extension of the body's thyroid hormone level, than free T4 itself. A person whose free T4 is at the very upper end of normal will have a TSH at the very lower end of normal. A person whose free T4 is at the lower end of normal will have a TSH at the upper end. These differences in TSH within the normal range are clinically meaningful. Patients tend to feel best and have optimal metabolic and cardiovascular markers at a specific TSH value that is personal to them, and small changes from this individual set point produce perceptible changes in well-being. The normal population range for TSH, approximately 0.4 to 4.0 μ/L, in most laboratory reference ranges, encompasses a 40-fold variation, reflecting the enormous individual variation in the free T4 level at which the hypothalamic pituitary system reaches its equilibrium. The TSH measures not absolute thyroid hormone, but whether the pituitary is getting what it considers the right amount, a personalized, rather than population-normalized hormonal signal. The discovery that bone is a mechanosensory endocrine organ, responding to the mechanical loading of physical activity by upregulating the production of osteocalcin, irisin from muscle, and other exercise-responsive hormones, reveals a hormonal conversation between the skeleton and the metabolic tissues that underlies many of the systemic health benefits of physical activity. The exercise-responsive hormones constitute a category of endocrine signals that was barely recognized 2 decades ago, and has since been found to mediate many of the health benefits of physical activity that were previously attributed to purely mechanical or metabolic mechanisms. Irisin, a peptide cleaved from the membrane protein FNDC5 by proteolytic processing, released into the bloodstream by exercising muscle, acts on white adipose tissue to promote its conversion to metabolically active brown-like adipose tissue, increasing thermogenesis and energy expenditure. It acts on bone to promote osteoblast activity, contributing to the bone-strengthening effects of exercise. And it acts on the brain, particularly the hippocampus, to promote neurogenesis and to improve cognitive function, providing one of the hormonal pathways through which exercise improves brain health. Osteocalcin, as already described, rises during exercise and enhances muscle performance. Meteorin-like protein, produced by muscle and adipose tissue during exercise, promotes anti-inflammatory programming in adipose tissue macrophages, contributing to the anti-inflammatory effect of regular physical activity. The collective effect of these exercise-responsive hormones, acting on fat, bone, brain, and immune system, provides a hormonal explanation for the extraordinary breadth of exercise's health benefits, from reduced cardiovascular disease and cancer risk, through improved cognition and mental health, to reduced inflammatory markers and improved metabolic health. Exercise is, in this hormonal view, a systemic signaling event, the working muscles and bones broadcasting a chemical message to every organ in the body that physical effort is underway and metabolic adaptation is required. The gastric bypass surgery and its related bariatric procedures produces dramatic improvements in type 2 diabetes within days of surgery, long before significant weight loss has occurred, through hormonal mechanisms that include changes in GLP-1, GIP, and other gut hormone secretion patterns that restore beta cell function and reduce insulin resistance independently of weight change. The observation that gastric bypass surgery improved type 2 diabetes almost immediately after the operation, before patients had lost more than a few percent of their body weight, was one of the most significant and most practically important discoveries in metabolic medicine of recent decades. It established unambiguously that the metabolic benefits of bariatric surgery were not simply the consequence of weight loss, and it pointed toward hormonal mechanisms emanating from the gut. The Roux-en-Y gastric bypass reroutes the intestinal tract so that food passes directly from a small gastric pouch to the jejunum, bypassing the duodenum and the proximal jejunum. This anatomical rearrangement dramatically changes the pattern of gut hormone secretion. Food that previously stimulated duodenal K cells to produce GIP now bypasses those cells, while the rapid delivery of nutrients to the more distal L cells of the ileum produces an exaggerated GLP-1 response after every meal. The hyperphysiological GLP-1 peaks following surgery produce powerful insulin secretion, suppress glucagon, slow gastric emptying, and strongly suppress appetite. The combination of these effects, normalized postprandial glucose excursions, improved beta cell function, reduced appetite, and eventually substantial weight loss, produces the remission of type 2 diabetes that is observed in approximately 50 to 60% of gastric bypass patients. The evidence that these hormonal changes occur before significant weight loss established the gut hormones as therapeutic targets in their own right, contributing to the urgency with which GLP-1 receptor agonists were developed as pharmacological tools. The hormonal regulation of appetite involves a complex hierarchy, from hypothalamic circuits integrating leptin, ghrelin, and many other signals at the top, through brainstem satiety circuits receiving vagal and hormonal inputs, to the hedonic circuits of the reward system that modulate the palatability and motivational significance of food beyond its caloric content. Appetite regulation is not a single system, but a hierarchy of systems operating at different levels of the brain, each integrating different signals and governing different aspects of food-motivated behavior. At the apex of the hierarchy is the hypothalamic arcuate nucleus, the primary homeostatic feeding center, where opposing populations of neurons translate the body's energy state into appetite and satiety signals. The neuropeptide Y and agouti-related protein neurons, the orexigenic circuit, are activated by ghrelin, by falling leptin, by low glucose, and by many other signals of energy deficit, and they drive food seeking and meal initiation. The pro-opiomelanocortin neurons, the anorexigenic circuit, are activated by leptin, by insulin, by many satiety hormones, and by high glucose, and they suppress appetite and increase energy expenditure. The nucleus of the solitary tract in the brainstem receives the vagal signals from the gut, the mechanosensory stretch signals of gastric distension, the chemosensory signals from enteroendocrine cells, the circulating satiety hormones, and integrates them into a brainstem satiety signal that suppresses meal intake. And the hedonic system, the dopaminergic reward circuits of the ventral tegmental area, and the nucleus accumbens, governs the palatability and motivational value of food, determining not just how much is eaten but what is eaten, under the influence of specific food cues, stress, social context, and the history of previous food rewards. The appetizing quality of dessert after a filling meal, the loss of appetite during grief, the stress eating that comfort food provides. These are the hedonic system operating over, and sometimes against the homeostatic system's signals. The concept of hormonal imprinting, the ability of hormones present during critical developmental windows to permanently program the sensitivity of the developing tissues to hormonal signals in adult life, reveals the endocrine system's participation in the developmental origins of health and disease. The Barker hypothesis, now called the developmental origins of health and disease, or DOHD, proposed in the 1990s that the nutritional environment of the fetus and neonate programs the physiology of the adult in ways that can increase or decrease the risk of metabolic, cardiovascular, and other diseases across the lifespan. The hormonal component of this programming involves the permanent alteration of the sensitivity of developing tissues to hormonal signals by the hormonal environment of the critical developmental window. Glucocorticoid exposure in utero, whether from maternal stress, from placental insufficiency, or from pharmacological administration, programs the HPA axis of the offspring toward a hyperreactive stress response that persists into adult life. The mechanism involves glucocorticoid-induced changes in the epigenetic programming of the HPA axis, the methylation and acetylation patterns of the glucocorticoid receptor gene, and of the genes in the feedback circuitry that are maintained through the divisions of the cells that carry them. The fetal pancreatic beta cells, programming their functional capacity in response to the nutritional environment of the womb, develop differently in the context of maternal diabetes, with implications for the offspring's own insulin secretory capacity and diabetes risk. The fetal hypothalamic circuits governing metabolic set points are programmed by the nutritional and hormonal environment of gestation, establishing the metabolic trajectory of the adult, long before the adult environment has any influence. The adult body is, in part, the hormonal legacy of the intrauterine environment. The role of sex hormones in the development of secondary sexual characteristics at puberty, the breast development and fat redistribution of estrogen, the muscle development and facial hair and voice deepening of testosterone, the growth spurt driven by both sex steroids and growth hormone, represents the most visible endocrine transformation of the lifespan. Puberty is the endocrine system's most dramatic performance, the transformation of the child's body into the adult's body, over the course of 3 to 5 years, driven by the reactivation of the hypothalamic GnRH pulse generator, and the consequent rise of sex steroids that had been suppressed since early infancy. In girls, the typical sequence begins with breast development, thelarch, driven by the rising estradiol from the developing ovarian follicles, followed by pubic hair growth driven partly by adrenal androgen, adrenarche, and partly by ovarian androgen, followed by the growth spurt, driven by the combination of GH and sex steroids, and finally by the onset of menstruation, menarche, which typically occurs approximately 2 to 2.5 years after the start of breast development, and signals the completion of the hypothalamic pituitary ovarian axis's maturation. In boys, the typical sequence begins with testicular enlargement, the earliest and most reliable sign of puberty, reflecting the response of the Leydig cells to rising LH, followed by pubic hair growth, penile growth, and then the growth spurt, which occurs approximately 2 years later in boys than in girls, partly explaining the height advantage that adult males eventually acquire over adult females despite similar childhood heights. The growth spurt itself is mediated by both the sex steroids and GH, the sex steroids amplifying GH secretion and increasing the sensitivity of the growth plate to IGF-1. And it is also the sex steroids that eventually close the growth plates by the end of puberty. The orchestration of these changes across 4 to 5 years, in a sequence that is broadly conserved across the species, but individually variable in its timing, is a demonstration of the endocrine system's capacity for sustained, coordinated developmental programs of extraordinary complexity. The connection between the endocrine system and the circadian clock is bidirectional. Hormones regulate the peripheral circadian clocks in many tissues, and the circadian clocks regulate the timing of hormone synthesis and secretion, creating a system in which temporal coordination is embedded in both the hormonal signals and their targets. The molecular circadian clock, the transcription translation feedback loop involving clock, BMAL1, period, and cryptochrome proteins, runs not only in the suprachiasmatic nucleus master clock, but in virtually every cell in the body, including the cells of every endocrine gland. These peripheral clocks govern the timing of the biochemical processes within each gland, producing the circadian rhythms and hormone synthesis and secretion that characterize each axis. the adrenal gland's own clock contributes to the circadian pattern of cortisol secretion, ensuring that the adrenal's sensitivity to ACTH is highest in the early morning, when the cortisol rise is needed. The testicular clock regulates the timing of testosterone synthesis. The liver clock, one of the most robust peripheral clocks in the body, coordinates the timing of metabolic processes, including insulin sensitivity and glucose production, in anticipation of the expected feeding patterns of the day. Hormones also set the timing of peripheral clocks through direct effects on the clock genes. Cortisol, for instance, acts as a strong synchronizer of peripheral clocks through glucocorticoid response elements and the promoters of clock genes. Insulin and feeding cues synchronize the liver and adipose tissue clocks. The practical consequence of this bidirectional relationship is that disruption of the circadian system by shift work, jet lag, irregular sleeping and eating times disrupts not just the suprachiasmatic clock, but the entire hormonal temporal architecture, producing the hormonal dysregulation that underlies the metabolic and reproductive consequences of circadian disruption. The endocrine system does not merely change with time. It is the mechanism by which the body knows what time it is. The concept of hormesis in endocrinology, the phenomenon in which low doses of a hormone or stressor produce beneficial adaptive responses while high doses produce harm, as informed understanding of cortisol, sex steroids, and exercise-responsive hormones, and has contributed to the emerging science of optimal hormonal environments. Hormesis is the dose-response relationship that violates the simple expectation of linear harm. At low or moderate doses, a stressor or a hormone produces beneficial adaptive responses. At high doses, it produces harm. Cortisol is the classic hormetic hormone in the endocrine system. At basal physiological levels, cortisol is essential. Its absence, in adrenal insufficiency, is life-threatening. At the moderate levels of the normal stress response, cortisol is adaptive, mobilizing energy, modulating inflammation, maintaining vascular tone and alertness. At the high levels of chronic stress or pharmacological dosing, cortisol is harmful, producing muscle wasting, immune suppression, hippocampal damage, metabolic dysfunction. The hormetic window, the range of cortisol exposure that produces benefit rather than harm, is the range in which the normal HPA axis operates in health. Sex steroids show similar hormetic patterns. Testosterone at physiological levels in men promotes muscle strength, bone density, mood, and cognitive function. At supraphysiological levels, pharmacological doping, it produces cardiovascular harm, testicular atrophy, and psychological disturbance. The exercise-responsive hormones, irisin, FGF21, osteocalcin, are produced in proportion to the exercise stimulus, with the beneficial metabolic effects observed at the moderate levels, associated with regular moderate exercise, and potential adverse effects if exercise becomes excessive, and the hormonal environment becomes one of chronic stress rather than adaptive stimulus. The recognition of hormesis in endocrinology has shifted the framing from simply more hormone equals more effect to the recognition that optimal hormonal levels exist within windows that are narrow enough to require respect. The measurement of hormones in saliva rather than blood has expanded the practical accessibility of endocrine testing because salivary hormone concentrations, particularly of cortisol, testosterone, estradiol, and progesterone, closely reflect the free, biologically active fraction in the blood, and salivary collection can be performed non-invasively at home, at any time of day. The clinical and research application of salivary hormone measurement has transformed the accessibility of endocrine investigation, particularly for hormones that must be measured at multiple time points or in natural settings outside a clinical environment. The basis for salivary hormone measurement is the diffusion of free, unbound steroid hormones across the salivary gland epithelium from the plasma. Only the free fraction, not the protein-bound fraction, is able to enter the saliva, which makes salivary concentration a direct reflection of the biologically active free hormone in the blood. For cortisol, whose circadian rhythm spans multiple time points across the day, and whose cortisol awakening response requires sampling within the first hour of waking, salivary collection has been transformative. Subjects can collect timed saliva samples at home without venipuncture, providing a more ecologically valid and less invasive measure of the cortisol awakening response in the diurnal cortisol slope. For research on testosterone's relationship to behavior, competition, and social dynamics, salivary sampling before and after competitive events has enabled studies that would have been impossible with venipuncture. The limitations of salivary measurement include the need to avoid food, drink, and oral hygiene within 30 to 60 minutes of collection, and the variability introduced by salivary flow rate and contamination. But for many endocrine applications, particularly the sampling of circadian patterns, the non-invasive study of hormonal correlates of behavior, and the monitoring of treatment and hormone replacement therapy, salivary measurement has provided a window into the hormonal system that was previously available only through clinic visits and venipuncture. The concept of the endocrine microenvironment, the local hormonal conditions within specific tissues, that can differ substantially from the systemic circulating hormone levels, is exemplified by the intratumoral estrogen production of breast cancer, the intraprostatic androgen concentrations maintained by local steroidogenesis, in castration-resistant prostate cancer, and the paracrine growth factor milieu of the bone metastatic niche. The hormone levels measurable in the blood provide the systemic picture of the endocrine environment, but many tissues maintain their own local hormonal milieu through local synthesis, local activation, or local concentration of hormones in ways that cannot be inferred from circulating levels. The breast is an example. Aromatase enzyme expressed in the breast stroma and in breast cancer cells converts androgens to estrogens locally, producing intratumoral estrogen concentrations substantially higher than the systemic estrogen levels. This local estrogen production drives the growth of estrogen receptor-positive breast cancers, even in postmenopausal women with very low systemic estrogen, which is why aromatase inhibitors, drugs that block the aromatase enzyme throughout the body, are more effective than treatments that simply reduce systemic estrogen in postmenopausal breast cancer. The prostate maintains high intraprostatic androgen concentrations through local synthesis from adrenal precursors and through efficient uptake and metabolism of circulating androgens, concentrations 10 times higher than in the blood. In castration-resistant prostate cancer, this intraprostatic androgen synthesis continues even when systemic testosterone has been reduced by castration, driving tumor growth. The clinical implication is that castration-resistant prostate cancer must be treated not only by reducing systemic androgen, but by suppressing the intraprostatic androgen synthesis through drugs like abiraterone, which blocks CYP17A1 throughout the body. The endocrine microenvironment of the tumor is, in these cases, as important as the systemic hormonal environment for determining tumor behavior and response to therapy. The hormonal changes of pregnancy, rising HCG, progesterone, estrogen, human placental lactogen, relaxin, and many others, produce the adaptations that accommodate the metabolic, cardiovascular, and immunological demands of gestation, including the expansion of blood volume by approximately 40%, the increased insulin resistance of the third trimester, and the immune tolerance of the semi-allogeneic fetus. Pregnancy is, from the endocrine system's perspective, a period of the most profound and most rapid hormonal transformation of adult life. A 9-month sequence of hormonal changes more dramatic than any other transition except perhaps puberty. The rising HCG of the first trimester maintains the corpus luteum and prevents menstruation. The progesterone that the corpus luteum and then the placenta produce relaxes the uterine smooth muscle, maintaining pregnancy. The rising estrogen of the second and third trimesters drives the growth of the uterus and the breast, expands the blood volume by increasing red cell mass and plasma volume, and prepares the breast ducts and alveoli for lactation. Human placental lactogen, a growth hormone-like peptide produced by the placenta, in quantities that exceed the pituitary's own growth hormone output by the third trimester, promotes insulin resistance in maternal tissues, diverting nutrients toward the fetus. The insulin resistance of late pregnancy, which can reveal an underlying predisposition to type 2 diabetes, as gestational diabetes in approximately 10% of pregnancies, is the maternal sacrifice of metabolic efficiency for fetal nutrient supply. Relaxin, produced by the corpus luteum and the placenta, relaxes the ligaments of the pelvis in preparation for delivery. And the immune tolerance of the fetus, which carries paternal antigens that would normally trigger an immune response, is maintained by a complex hormonal and immunological program involving progesterone, HCG, and placental immune modulators that has fascinated reproductive immunologists for decades. Pregnancy remakes the hormonal body as thoroughly as any process in biology. The seasonal variation in hormone levels, testosterone and FSH showing modest seasonal patterns in many populations, vitamin D levels varying substantially with latitude and season, and melatonin patterns shifting with day length, reveals the endocrine system's historical responsiveness to the annual light cycle that shaped its evolution in seasonally variable environments. The endocrine system of modern humans retains vestiges of the seasonal hormonal programming that was essential for the reproductive and metabolic adaptation to the annual cycle, in environments with dramatically different summer and winter conditions. In many mammals, reproduction is strictly seasonal, timed by melatonin's seasonal signal to the hypothalamic pituitary gonadal axis, to coincide with the optimal environmental conditions for offspring survival. In humans, reproduction is not strictly seasonal, but subtle seasonal influences remain. Testosterone shows modest seasonal variations in some population studies, with some reporting slightly higher levels in autumn. Birth rates in many populations show modest seasonality that may reflect a combination of seasonal variation in sexual activity and modest seasonal variation in fertility. Vitamin D shows the most dramatic seasonal variation, dependent on ultraviolet B radiation for its synthesis in the skin. It falls to deficient levels in winter in many temperate populations, with consequences for immune function, mood, and bone health. The shorter days and longer nights of winter produce longer nocturnal melatonin secretion and the extended dark phase signal that drives seasonal reproductive quiescence in strictly seasonal mammals. In humans, the same signal may contribute to the seasonal affective disorder that affects approximately 2 to 3% of northern populations in the winter months, producing the depression, carbohydrate craving, and hypersomnia that represent a mild expression of the winter adaptive program. The endocrine system remembers the world that shaped it, even in the worlds that have moved beyond that shaping. The hormonal response to cold exposure, including the sympathetic stimulation of brown adipose tissue thermogenesis, the shivering-induced release of irisin and FGF21, and the activation of the HPA axis, reveals the endocrine system's essential role in maintaining the core body temperature that the enzymes of metabolism require. Thermoregulation is a fundamental endocrine task. The body's metabolic rate, and therefore its heat production, is determined in large part by thyroid hormone, which sets the rate of cellular respiration in all tissues. But acute cold exposure requires an immediate thermogenic response, faster than thyroid hormone's genomic action can provide, and the endocrine system has evolved multiple rapid thermogenic mechanisms. The sympathetic nervous system activates immediately upon cold exposure, stimulating the brown adipose tissue's thermogenic program through beta-3 adrenergic receptors. Brown adipose tissue, containing densely packed mitochondria whose inner membrane is rich in uncoupling protein 1, responds by decoupling oxidative phosphorylation from ATP synthesis, converting the energy of substrate oxidation directly to heat rather than to ATP. The result is a thermogenic organ that can, when fully activated, produce several hundred watts of heat per kilogram of tissue. In adults, brown adipose tissue is concentrated in the supraclavicular region, the perirenal region, and around the spinal cord, and its mass and thermogenic capacity increase with cold acclimatization. Shivering, the coordinated, heat-generating oscillation of skeletal muscle, is also a cold-responsive thermogenic mechanism, and the muscular effort of shivering produces the myokines irisin and FGF21 that, as noted, have effects on adipose tissue browning and metabolic activation. The HPA axis is activated by cold through thermosensory pathways, and the rising cortisol of cold exposure provides the metabolic substrate mobilization to fuel the thermogenic effort. The body maintains its temperature through a hormonal and neural program of remarkable coordination, because the enzymes of cellular life, including those that produce the hormones we have been discussing, operate only within a narrow thermal range. The insulin receptor, a transmembrane tyrosine kinase activated by insulin binding to its two extracellular alpha subunits, initiates a signaling cascade through IRS proteins, PI3 kinase, ACT, and mTOR that represents one of the best characterized intracellular signaling pathways in biology, and has yielded multiple drug targets for metabolic disease. The insulin receptor is a tetrameric structure consisting of two extracellular alpha subunits and two transmembrane beta subunits, held together by disulfide bonds. When insulin binds to the two alpha subunits, with a high affinity in the nanomolar range that reflects the physiologically relevant concentrations of circulating insulin, the receptor undergoes a conformational change that activates the intrinsic tyrosine kinase activity of the two beta subunits, which transphosphorylate each other and then phosphorylate the cytoplasmic adapter proteins called insulin receptor substrate proteins, IRS1 through IRS4. Phosphorylated IRS proteins serve as docking sites for PI3 kinase, phosphatidylinositol 3 kinase, which generates the lipid second messenger PIP3 at the inner membrane surface. PIP3 recruits and activates the serine threonine kinase ACT, also called protein kinase B, which is the central effector of the metabolic actions of insulin. ACT phosphorylates and inhibits glycogen synthase kinase 3, allowing glycogen synthase to be active and promoting glycogen synthesis. It phosphorylates and inhibits the fork head transcription factors, suppressing the gluconeogenic enzyme program. And in muscle and fat cells, it promotes the translocation of GLUT4 vesicles to the plasma membrane, the critical step in insulin-stimulated glucose uptake. The mTOR complex, activated downstream of ACT, stimulates protein synthesis. This pathway, from the insulin receptor through IRS to PI3K to ACT to the metabolic effectors, is the substrate of the most successful pharmacological targeting in type 2 diabetes treatment, with drugs targeting multiple steps from GLP-1-mediated receptor amplification to SGLT2-mediated renal glucose excretion, and with the IRS-PI3K ACT pathway itself under investigation as a target for the treatment of insulin resistance. The hormone melatonin acts not only as the body's circadian phase signal, but as a potent antioxidant directly and through stimulating the expression of antioxidant enzymes, with its nighttime production coinciding with the period of greatest cellular repair, and its mitochondrial uptake providing local antioxidant protection at the site of the greatest reactive oxygen species generation. Melatonin's antioxidant properties were first described in the early 1990s, when it was found to directly scavenge hydroxyl radicals with an efficiency exceeding that of the classical antioxidants vitamin C and vitamin E. Subsequent work showed that melatonin does not merely scavenge radicals directly, but also stimulates the expression of the antioxidant enzymes superoxide dismutase, catalase, and glutathione peroxidase, enzymes that form the front line of the cellular defense against reactive oxygen species. The coincidence of melatonin's nightly rise with the body's period of greatest cellular repair, the deep sleep during which the brain's glymphatic system clears metabolic waste, the skin divides and renews, and growth hormone drives tissue anabolism, suggests that the antioxidant protection of melatonin may be part of the repair program, protecting the replicating DNA of dividing cells from oxidative damage during the period when cellular division is most active. Melatonin's entry into mitochondria, the primary site of reactive oxygen species production in the cell, is of particular interest, as it provides antioxidant protection precisely where the oxidative burden is greatest. The decline of melatonin production with age, part of the age-related involution of the pineal gland, has been proposed as a contributor to the increased oxidative stress of aging, and the age-related loss of the nightly antioxidant protection that melatonin provides. Tonight's melatonin is not only the signal to sleep, it is also, quietly, the night shift of cellular protection. The connection between the endocrine system and the microbiome, the community of microorganisms inhabiting the gut, involves bidirectional influences in which gut bacteria metabolize and activate certain hormones, produce molecules that activate hormone receptors, and are themselves regulated by hormonal signals, creating a microbiome endocrine axis whose disruption may contribute to metabolic disease. The gut microbiome, the approximately 38 trillion bacteria, archaea, fungi, and viruses inhabiting the human gastrointestinal tract, is an ecological community that has co-evolved with the human host and participates in the host's physiology in ways that are only beginning to be fully characterized. The endocrine intersection is both direct and indirect. Directly, the gut bacteria metabolize estrogens and other steroid hormones excreted in the bile, converting them back to forms that can be reabsorbed and recirculated. The enterohepatic circulation of estrogen. The beta-glucuronidase activity of certain gut bacteria deconjugates the conjugated estrogens excreted by the liver, releasing free estrogens that can be reabsorbed in the colon, extending the effective half-life of estrogen and influencing the systemic estrogen level. Disruption of the gut microbiome by antibiotics, for instance, can alter this enterohepatic circulation and change systemic estrogen levels. The gut bacteria also produce short-chain fatty acids, butyrate, propionate, acetate, that act on G protein-coupled receptors in the gut epithelium, the liver, and adipose tissue, modulating insulin sensitivity, fat storage, and appetite regulation. Propionate activates GPR41 and GPR43 receptors on the gut enteroendocrine cells, stimulating the release of PYY and GLP-1. Butyrate provides the primary energy source for the colonocytes and has epigenetic effects on gene expression throughout the gut epithelium. The microbiome, in this sense, is a biochemical extension of the endocrine system, a community of organisms whose metabolic outputs are hormonal signals and hormonal regulators, integrated into the host's endocrine physiology through millions of years of coevolution. The phenomenon of sex hormone-binding globulin as a dynamic regulator of sex steroid bioavailability reveals how the endocrine system uses protein binding not merely as a passive transport mechanism but as an active regulatory system, with insulin suppressing SHBG production and thereby increasing free testosterone in conditions of hyperinsulinemia. The production of SHBG by the liver is not constitutive and fixed but regulated by a variety of hormonal and metabolic signals that allow the liver to adjust the bioavailability of sex steroids in response to the body's overall metabolic and hormonal context. Insulin suppresses SHBG production. The hyperinsulinemia of insulin resistance and type 2 diabetes is associated with low SHBG levels, which increases the free fraction of testosterone and estradiol. This is one of the mechanisms contributing to the hyperandrogenism of polycystic ovary syndrome, where hyperinsulinemia drives SHBG suppression, increasing the free androgen fraction and exacerbating the androgen excess that characterizes the condition. Estrogen, and oral contraceptives containing estrogen, increases SHBG production, reducing the free androgen fraction, which is one mechanism by which combined oral contraceptives can treat the androgenic symptoms of polycystic ovary syndrome. Androgens and growth hormones suppress SHBG, thyroid hormones, some glucocorticoids, and certain liver conditions increase it. The resulting SHBG level at any given time is the hepatic summary of the hormonal and metabolic environment, a readout of the combined influences of multiple hormones on a single regulatory protein that determines the bioavailability of the sex steroids throughout the body. Measuring SHBG alongside total testosterone and estradiol provides information that cannot be obtained from either measurement alone, and calculating the free hormone from the total and the binding protein is one of the more nuanced interpretive tasks in endocrine medicine. The adrenal incidentaloma. The adrenal nodule discovered incidentally on imaging performed for another indication has become one of the most common clinical dilemmas in endocrinology as CT scanning has become ubiquitous, because a substantial minority of these nodules produce small amounts of cortisol autonomously, causing a subclinical Cushing syndrome whose metabolic consequences include hypertension, diabetes, and osteoporosis. The adrenal incidentaloma is a product of modern medicine's imaging capabilities, a lesion that was rarely identified before the routine use of cross-sectional imaging and is now found in approximately 2 to 4% of abdominal CT scans performed for any reason. The majority of adrenal incidentalomas are benign, non-functioning cortical adenomas that produce no hormone in excess and require no treatment beyond surveillance. But approximately 10 to 15% secrete cortisol autonomously, not enough to produce the florid clinical syndrome of Cushing's disease, but enough to partially suppress the normal HPA axis negative feedback and to expose the body to mildly elevated cortisol that, over years, produces the metabolic consequences of glucocorticoid excess, visceral obesity, hypertension, insulin resistance, dyslipidemia, and reduced bone density. This subclinical Cushing syndrome was first clearly characterized in the 1990s and remains a clinical entity without universally agreed management guidelines. The debate persisting about whether surgical removal of the adenoma is preferable to continued surveillance in patients without dramatic clinical symptoms. The surgical argument draws on evidence that adrenalectomy in patients with autonomous cortisol secretion improves hypertension, glucose tolerance, and body composition. The surveillance argument notes that the evidence base is not sufficiently strong to recommend surgery in all cases. The adrenal incidentaloma has, in this sense, created a new category of endocrine disease, subclinical hormonal excess, that exists only because the imaging tools to detect it have become so widely available. The hormonal programming of sexual differentiation in the developing brain, distinct from but related to the hormonal programming of the genitalia, involves critical period exposure of hypothalamic and limbic neurons to androgens and estrogens that permanently shapes the neural circuits governing reproductive behavior, stress responses, and certain cognitive patterns. The sexual differentiation of the brain occurs later in fetal development than the sexual differentiation of the genitalia, and it is accomplished by different mechanisms. The genitalia are differentiated in the first trimester primarily by testosterone and its derivative DHT acting on the genital tissues. The brain is differentiated predominantly in the second trimester and in the early neonatal period, by a combination of testosterone acting directly on androgen receptors in the brain, and estradiol produced locally by aromatization of testosterone within brain cells acting on estrogen receptors. This seemingly paradoxical finding, that estrogen masculinizes the developing male brain, reflects the brain's different receptor ecology compared to the genitalia. In the brain, aromatase is abundantly expressed, and much of testosterone's masculinizing effect is through local estrogen. Female fetuses are protected from masculinization by alpha-fetoprotein, which binds estradiol in the fetal circulation and prevents it from crossing into brain cells, while the testosterone produced by the male testes enters brain cells directly and is locally converted to estradiol. The critical period for these brain masculinizing effects in humans spans roughly the second trimester of gestation and possibly the early neonatal period. The brain structures affected include the hypothalamic nuclei governing the reproductive hormone cycles, explaining why women have cyclical LH release and men do not, and limbic structures governing stress responsivity, certain aspects of spatial cognition, and aspects of social behavior. The pharmacological modulation of the endocrine system, through drugs that mimic, block, stimulate, or suppress hormonal signals, represents one of the most clinically impactful domains of medicine, with hormone therapies transforming the treatment of diabetes, hypothyroidism, contraception, menopause, cancer, and many other conditions. The endocrine system's reliance on specific molecular interactions, hormone binding to receptor activating signaling cascade, makes it unusually amenable to pharmacological targeting. The design of molecules that mimic the natural hormone, agonists, or that bind the receptor without activating it, antagonists, has produced therapeutic agents of enormous clinical importance across endocrinology and beyond. Exogenous insulin, first animal-derived, then biosynthetic human insulin, now designer analog insulins engineered for specific pharmacokinetic profiles, has extended the lives of hundreds of millions of people with diabetes. Levothyroxine, synthetic T4, replaces the thyroid function of the hundreds of millions worldwide with hypothyroidism. The oral contraceptive pill, combining synthetic estrogen and progestogen to suppress the LH surge and prevent ovulation, has transformed reproductive autonomy for hundreds of millions of women. The selective estrogen receptor modulators, tamoxifen and raloxifene, compete with estradiol for the estrogen receptor, acting as estrogen agonists in some tissues and antagonists in others, allowing the therapeutic exploitation of estrogen's tissue-specific beneficial effects while blocking its harmful ones. The aromatase inhibitors suppress the conversion of androgens to estrogens, reducing estrogen levels in postmenopausal breast cancer to near zero and improving survival. The glucocorticoids, prednisone, dexamethasone, hydrocortisone, suppress inflammation and immune activation in conditions ranging from asthma to lupus to organ transplantation. The breadth of endocrine pharmacology is, in a sense, the breadth of the endocrine system itself. Where hormones govern, drugs can follow. The discovery of new hormones continues in the 21st century, with recent additions including meteorin-like protein, neuregulin-4, FNDC5, lipocolin-2, and many others from unexpected tissues including muscle, bone, fat, and brain, suggesting that the endocrine map of the body is still far from complete and that the chemical conversation among tissues extends beyond what has yet been characterized. The history of endocrinology is the history of expanding the map, of recognizing, one by one, that tissues previously thought to be purely mechanical or structural were actually chemical communicators, secreting signals into the bloodstream or the local tissue environment that govern the function of other tissues at a distance. The heart with atrial natriuretic peptide, bone with osteocalcin, muscle with irisin and dozens of other myokines, fat tissue with leptin, adiponectin, and a growing list of adipokines. The gut with 20 or more hormones. The liver with FGF21, hepcidin, fetuin A. The brain with neuropeptides that enter the circulation and act on peripheral tissues. Each of these additions to the hormonal map was preceded by the assumption that the tissue in question was not an endocrine organ, and each assumption was wrong. The implication for the completeness of the current map is humbling. If so many tissues that were confidently classified as non-endocrine have turned out to be active hormonal secretors, the assumption that we have now identified all the significant hormones and all the significant hormonal sources requires considerable skepticism. The tools for discovering new secreted molecules and their receptors, proteomics, transcriptomics, CRISPR screens, are now powerful enough that new candidate hormones are being identified faster than their functions can be characterized. The endocrine system is, in all probability, substantially larger and more interconnected than any current model captures. The chemical conversation of the body is still being translated. Stay a while longer. You have stayed with me a very long time tonight. Through the hypothalamus and the pituitary and the thyroid and the adrenal glands and the pancreatic islets and the gut hormones and the adipokines and the endocrine heart and the hormonal bone. 100 facts about the chemistry that runs the body's long game. And you have been here for nearly all of them. I do not take that lightly. Time is the most honest gift there is. If relax with science has earned a small place in your evenings, if these nights have given you something you would like to return to, subscribing is the simplest and most honest way to say so. I will be here. I always will be. Just a little further. We are nearly where we are going. You have stayed with me a very long time tonight. Through the hypothalamus and the pituitary and the thyroid and the adrenal glands and the pancreatic islets and the gut hormones and the adipokines and the endocrine heart and the hormonal bone. 100 facts about the chemistry that runs the body's long game, and you have been here for nearly all of them. I do not take that lightly. Time is the most honest gift there is. If relax with science has earned a small place in your evenings, if these nights have given you something you would like to return to, subscribing is the simplest and most honest way to say so. I will be here. I always will be. Just a little further. We are nearly where we are going. The pituitary tumor, the adenoma arising from one of the 5 anterior pituitary cell types, is the most common intracranial tumor in adults, present in autopsy studies in approximately 10 to 20% of the general population, and produces its clinical effects either through the excess secretion of a specific hormone or through the mass effect on surrounding structures as it grows. The pituitary adenoma is one of medicine's most instructive endocrine conditions precisely because its effects are so directly predictable from knowledge of the specific cell type that has undergone autonomous proliferation and from knowledge of the anatomy of the pituitary fossa. A somatotroph adenoma, producing excess growth hormone, will produce acromegaly in an adult, the gradual enlargement of the hands, feet, and facial features, the soft tissue overgrowth, the metabolic consequences of GH excess including diabetes, and the cardiovascular complications that make untreated acromegaly a condition of significantly reduced life expectancy. A corticotroph adenoma, producing excess ACTH, which drives excess cortisol from the adrenal, will produce Cushing's disease, the characteristic central obesity with peripheral wasting, the moon face, the buffalo hump, the purple striae, the hypertension, the diabetes, the osteoporosis, the psychiatric disturbances. A prolactinoma, by far the most common secreting adenoma, will produce hyperprolactinemia, suppression of GnRH pulsatility with consequent hypogonadism, galacturia, and the visual field defect if the tumor is large enough to compress the optic chiasm, the crossing visual pathways that pass immediately above the pituitary. A thyrotroph adenoma will drive hyperthyroidism through excess TSH, and a gonadotroph adenoma, typically non-secreting in a clinically meaningful way, will present through mass effects on the adjacent structures. The pituitary fossa is one of the most anatomically constrained spaces in the body, surrounded by bone on 3 sides and by the optic chiasm above, and the expansion of a tumor within it produces the predictable sequence of hormonal effects and visual and neurological complications that make pituitary adenoma management one of the most multidisciplinary challenges in endocrinology. The chronobiology of the endocrine system reveals that the timing of hormonal administration, giving a medication at the biological time when its target tissue is maximally responsive, can dramatically improve therapeutic efficacy and reduce side effects, an emerging field called chronopharmacology. Chronopharmacology is the application of the endocrine system's temporal architecture to the design of drug dosing regimens. If a hormone receptor's sensitivity varies across the day, or if the signaling pathway downstream of a receptor shows circadian variation in its activity, then the therapeutic effect of a drug acting on that receptor will vary with the time of administration, even if the drug's pharmacokinetics are identical at different times of day. Glucocorticoid therapy provides the clearest clinical example. Synthetic glucocorticoids administered in the morning, when the HPA axis is naturally at its peak, produce less suppression of the HPA axis than the same dose administered in the evening, because the morning administration is less dissonant with the normal cortisol rhythm. Once daily evening dosing of glucocorticoids maximizes HPA suppression and is preferred when that suppression is the therapeutic goal, as in congenital adrenal hyperplasia, while morning dosing minimizes HPA suppression and is preferred when it is an undesired side effect. Statins, drugs that inhibit the enzyme HMG-CoA reductase in the cholesterol synthesis pathway, are most effective when taken in the evening, when cholesterol synthesis is at its circadian peak. Low-dose aspirin for cardiovascular protection is more effective when taken at bedtime, when its antiplatelet effect coincides with the circadian peak of platelet aggregability in the morning. Antihypertensive medications may have different efficacy depending on whether the patient's hypertension pattern is predominantly diurnal or nocturnal. The circadian architecture of the endocrine system means that the clock on the wall is as relevant to pharmaceutical dosing as the dose itself. The hormonal changes associated with aging, the decline of DHEA, growth hormone, IGF-1, sex steroids, and melatonin have been extensively studied as potential targets for interventions to extend health span. But the evidence that restoring youthful hormone levels in older adults produces a net benefit rather than a net harm has proven difficult to establish. The history of hormonal anti-aging therapy is a cautionary tale about the difference between restoring hormonal levels to their youthful range and restoring the health state associated with those levels in youth. The initial logic is seductive. As the body ages, hormone levels fall, and many features of aging resemble the symptoms of hormone deficiency. Restore the hormones, and perhaps the features of aging will reverse. The estrogen replacement that menopause requires to prevent the acute symptoms and the long-term skeletal and cardiovascular consequences, widely used in the decades following the development of menopausal hormone therapy, was found in the Women's Health Initiative trial to carry risks of breast cancer, cardiovascular events, and stroke that had not been apparent from observational studies. The subsequent reanalysis showed that risks varied substantially with the type of hormone, the route of administration, and the timing of initiation relative to menopause, establishing the concept of the timing hypothesis for hormone therapy. Growth hormone administration in healthy older adults with low GH levels improves body composition and reduces fat mass, but does not clearly improve functional capacity or reduce mortality, and produces side effects including fluid retention, joint pain, and possible increased cancer risk. Testosterone supplementation in older men with low testosterone improves sexual function, muscle mass, and bone density, but the cardiovascular safety has been uncertain and the evidence for effects on mortality and cognition is mixed. The difficulty is that the hormone levels of youth co-occur with many other features of the young biological state. The resilience, the cellular regenerative capacity, the genomic stability, that cannot be simply restored by replacing the circulating hormone molecules. The phenomenon of ectopic hormone production, the synthesis of hormones by tumors arising from tissues that do not normally produce those hormones, reveals the latent hormonal potential of differentiated tissues and produces some of medicine's most diagnostically challenging and clinically dramatic presentations. Ectopic hormone production occurs when a tumor expresses genes that are normally silenced in the differentiated cell type from which it arose, producing a hormone that the host tissue had no prior history of making. The most common and most clinically significant examples involve small cell lung cancer, which produces ACTH with sufficient frequency that Cushing's syndrome due to ectopic ACTH from a lung cancer is a distinct clinical entity, characterized by the rapid onset of severe hypercortisolism, often with prominent hypokalemia from the mineralocorticoid activity of very high cortisol levels, and often presenting before the lung tumor is clinically apparent. Other tumors producing ACTH include carcinoid tumors, pancreatic neuroendocrine tumors, and thymic carcinoids. Small cell lung cancer also produces antidiuretic hormone ectopically, the syndrome of inappropriate ADH secretion, producing hyponatremia through inappropriate water retention, is a perineoplastic complication with substantial morbidity. Certain ovarian tumors produce HCG, producing the symptoms of pregnancy including morning sickness in non-pregnant women. Non-islet cell tumors producing IGF-2, particularly large sarcomas and hepatocellular carcinomas, can produce hypoglycemia through the insulin-like effects of IGF-2 on glucose metabolism. The clinical recognition of ectopic hormone production requires the clinician to consider a malignant source for hormonal excess even in the absence of the classical pituitary, adrenal, or thyroid origin, and the investigation of an unexpected hormonal syndrome often leads to the early detection of a malignancy that would otherwise have been diagnosed at a later, less treatable stage. The role of the endocrine system in the regulation of the immune response extends to the profound immunosuppression that is required for maternal tolerance of the semi-allogeneic fetus, a tolerance maintained by hormonal, cellular, and molecular mechanisms whose partial failure may contribute to the preeclampsia and recurrent miscarriage of disordered reproductive immunology. The fetus is genetically distinct from the mother. It carries paternal antigens that would ordinarily trigger an immune response, and the mechanisms by which the maternal immune system is prevented from rejecting the fetal allograft while retaining the ability to fight infection represent one of the most intricate immunological achievements of mammalian biology. The hormonal contribution to this tolerance is substantial. Progesterone, produced first by the corpus luteum and then by the placenta, suppresses T-cell proliferation and promotes the shift from pro-inflammatory Th1 responses toward tolerance-promoting Th2 responses at the maternal-fetal interface. HCG promotes the expansion of regulatory T cells, immune suppressor cells that actively dampen immune responses, and suppresses natural killer cell activity at the decidua. The placenta itself expresses HLA-G, a non-classical HLA molecule, which interacts with inhibitory receptors on natural killer cells and T cells at the decidual surface, preventing them from attacking the trophoblast. Estrogen and prolactin both modulate the decidual immune environment toward tolerance. The disruption of this hormonal immune balance, whether by placental dysfunction, by insufficient regulatory T-cell expansion, or by an immune environment tilted toward Th1 responses, is implicated in the development of preeclampsia, in recurrent implantation failure, and in the idiopathic recurrent miscarriage that affects approximately 1% of couples attempting pregnancy. The gut microbiome influences estrogen metabolism through the estrobolome, the subset of gut bacteria expressing beta-glucuronidase that deconjugate estrogen metabolites excreted in the bile, allowing their reabsorption and recirculation, and disruption of this microbiome estrogen axis may contribute to estrogen-dependent diseases including breast cancer and endometriosis. The estrobolome is the metabolic ecosystem within the gut microbiome that collectively manages the enterohepatic circulation of estrogens. The liver conjugates estrogens, attaching glucuronic acid to make them water-soluble, and excretes the conjugates in bile. In the colon, gut bacteria expressing beta-glucuronidase deconjugate these estrogen glucuronide conjugates, releasing free estrogens that can be reabsorbed from the colon and re-enter the systemic circulation. The diversity and composition of the beta-glucuronidase-expressing microbiome determines the efficiency of this deconjugation. A high-activity estrobolome recirculates more estrogen, contributing to higher systemic estrogen levels. A low-activity estrobolome allows more estrogen to be excreted in the feces, producing lower systemic estrogen. The clinical significance of this microbiome regulation of estrogen bioavailability has been explored in the context of estrogen-dependent cancers. Women with higher diversity microbiomes, with more robust estrobolome activity, may have higher systemic estrogen levels and potentially different breast cancer risk profiles. Antibiotic disruption of the gut microbiome reduces beta-glucuronidase activity and reduces estrogen reabsorption, an effect that has been proposed as one mechanism by which antibiotic use may potentially alter the efficacy of estrogen-containing oral contraceptives. The estrobolome is, in this sense, a microbiome endocrine interface that represents one of the most direct and best characterized examples of how the gut bacterial community participates in the hormonal regulation of the host. The regulation of appetite and body weight involves not only the homeostatic hormonal signals of leptin, ghrelin, and insulin, but also the allostatic system, the chronic upward or downward adjustment of the weight set point in response to prolonged changes in energy balance, which is the reason why weight loss is so difficult to maintain and why the body defends a higher weight following obesity. The set point theory of body weight regulation holds that the body defends a specific body weight, its set point, through adaptive changes in metabolic rate, appetite, and energy expenditure that oppose any deviation from that set point. Weight gain triggers satiety signals and metabolic suppression that resist further gain. Weight loss triggers hunger signals and metabolic adaptation that resist further loss. The set point is not fixed but is subject to upward drift, a process called allostasis. In the context of prolonged positive energy balance, poor sleep, chronic stress, and aging. The hormonal mechanisms of this set point defense have been extensively characterized: the post-weight loss rise in ghrelin, the post-weight loss fall in leptin, the reduction in peptide YY, the reduction in CCK, and the reduction in metabolic rate that together produce the persistent increased hunger and reduced energy expenditure that make weight maintenance after intentional weight loss so difficult. These hormonal changes persist for at least 12 months after weight loss and probably much longer, the body treating the weight loss as a deficit to be corrected rather than a goal to be maintained. The recognition that these hormonal defenses are a normal biological response to weight loss, and not a failure of willpower, has been one of the most important conceptual advances in obesity medicine and has driven the development of pharmacological and surgical approaches that can override these defenses by providing sustained artificial satiety signals through GLP-1 receptor agonism or by altering the gut hormone milieu through bariatric surgery. The hormonal changes of the postpartum period, the dramatic fall in estrogen, progesterone, and human placental lactogen at delivery, combined with the rise of prolactin and oxytocin for lactation, create the most abrupt hormonal transition of adult life and a vulnerability window for mood disorders including postpartum depression and postpartum psychosis. Delivery is, from the endocrine system's perspective, a hormonal cliff edge. The placenta, which has been producing estrogen, progesterone, HCG, and human placental lactogen in increasingly large amounts through the third trimester, is expelled within minutes of delivery, and the circulating levels of its hormones fall within hours from their peak pregnancy values to near zero. This abrupt withdrawal of estrogen, from the high levels of late pregnancy to the near castrate levels of the early postpartum period, before the ovarian production resumes, is the most rapid large magnitude estrogen drop that occurs in normal female physiology. The simultaneous withdrawal of progesterone, and therefore of its neuroactive metabolite allopregnanolone, which had been providing anxiolytic and sedative effects throughout pregnancy, removes an important neural buffering system. The rise of prolactin for lactation and the frequent release of oxytocin during breastfeeding provide some compensatory neural support. Both hormones have mood-supporting effects, but they are not equivalent to the hormonal environment of the last trimester. Postpartum depression, affecting approximately 10 to 15% of women in the weeks and months following delivery, is one of the most common serious complications of childbirth, and its hormonal basis is now clearly recognized in the fall of estrogen and allopregnanolone and the dysregulation of the HPA axis in the postpartum period. Brexanolone, the synthetic allopregnanolone approved for postpartum depression, provides direct evidence that the neurosteroid withdrawal is a meaningful contributor, because its administration produces rapid and dramatic improvement in a condition that until recently had no specific pharmacological treatment. The hormonal system's response to light, from the melatonin suppression of bright light to the vitamin D synthesis of ultraviolet exposure to the cortisol awakening response triggered partly by light, reveals the ancient solar programming of the endocrine system, adapted over millions of years to the predictable daily and annual cycles of the sun. The sun is the endocrine system's oldest signal. Long before any receptor in any organ was specialized for any other environmental input, the biological clocks of living organisms were calibrated to the 24-hour rotation of the Earth and the annual revolution around the sun. In the human endocrine system, this solar programming runs through multiple pathways. The retinal melanopsin cells, the intrinsically photosensitive retinal ganglion cells that provide the circadian light input, are maximally sensitive to the blue wavelengths of morning sunlight, providing the most potent Zeitgeber for the suprachiasmatic nucleus and the most potent suppressors of melatonin. Morning light exposure is the most effective means of advancing the circadian clock, and its regular use is the first-line treatment for delayed sleep phase disorder, the circadian timing problem in which the clock is set too late for conventional social schedules. The ultraviolet B of midday sunlight provides the photons that begin the vitamin D synthesis in the skin, a solar gift whose physiological importance is reflected in the broad tissue distribution of the vitamin D receptor and the diverse consequences of its deficiency. The cortisol awakening response is amplified by light exposure. Morning bright light activates the HPA axis more effectively than darkness, making the morning cortisol rise part of the body's photic entrainment as well as its anticipatory preparation for the day. The endocrine system learned the sun's language before it learned any other. Tonight, in the darkness, it is using what it learned. The melatonin is rising because it is dark, and it is dark because the earth has turned, and the earth turns as it has always turned, and the endocrine system keeps time. The endocrine system is, at its most fundamental, the biology of becoming, the chemical system through which the body is continuously remade, continuously adapted, continuously responding to what the world presents and what the body needs. And to understand it is to understand that every cell in your body is, at this moment, receiving a message from somewhere else in your body, a chemical message about what time it is, and what the body has eaten, and what challenges have arisen, and what rest is now required. Begin with the simplest version of this last fact. You are soaking in chemistry right now. Not metaphorically. The blood that is moving through the vessels of your body at this moment carries dozens of molecular messages. The melatonin of the pineal gland that is rising with the deepening dark, telling every cell in the body that it is night. The low cortisol of the late evening that is removing the wakefulness signal and permitting the sleep centers of the brain to assert themselves. The insulin that is managing whatever glucose remains from the last meal, directing it to where it needs to go. The growth hormone that is gathering itself for the pulse that will come when deep sleep begins to oversee the night's repair. The leptin from the fat tissue, speaking to the hypothalamus about the adequacy of the energy stores. The sex steroids at whatever level the day's demands and the reproductive season have set them. The thyroid hormones, steady and persistent, setting the metabolic rate of every cell in the body, the rate at which each cell burns its fuel, does its work, maintains its existence, against the entropy that is always pressing in. All of this is in the blood right now. All of this is in your blood right now. The chemistry of the endocrine system is not something that happens to the body. It is the body happening, the body's continuous chemical self-conversation, its internal government, its distributed intelligence about what is and what is needed and what to do. You have been that conversation all your life. Tonight, in the quiet, the conversation is finding its slower register. The message is being sent right now. Melatonin, low cortisol, the preparing pulse of growth hormone are the messages of rest. The endocrine system is speaking to itself in the language of the night. In the night, as it always does, is listening. The endocrine system you have been traveling through tonight is not separate from you. It is not a subject held at scholarly distance, examined from the outside. Every hormone discussed tonight is either in your bloodstream right now, or was earlier today, or will be before morning. The melatonin that is rising as you lie here. The growth hormone that will peak in the slow wave sleep that is approaching. The cortisol that reached its low point hours ago and will begin its slow climb back toward tomorrow's awakening. The insulin that settled the chemistry of the last meal. All of it yours. All of it already happening in the blood that moves through the same veins it has always moved through, carrying the same molecular messages it has always carried, in the same language that the body has been reading since before it was a body as we know it. The slowness of the endocrine system, the minutes it takes for a hormone to reach its target, the hours it takes for a steroid to change gene expression, the days it takes for thyroid hormone levels to stabilize after a dose adjustment, is not a limitation. It is the timescale of the body's deep life. The things the endocrine system governs: growth, metabolism, reproduction, the response to sustained challenge, the long-term calibration of mood and energy and resilience, are not things that happen in a second. They are things that happen across hours, and days, and years, and lifetimes. The endocrine system is keeping time in units that the nervous system cannot reach, and that the will cannot shortcut. It is the body's memory of what the world has been doing to it, and its plan for what to do in response. Tonight, as you rest, it is doing the quiet work of the dark hours, repairing, consolidating, calibrating, preparing the chemistry for tomorrow, and you do not need to be aware of it for it to be doing this work. You only need to be here. This is Relax with Science, saying goodnight and meaning it with the full measure of everything we have moved through together. The wonder of the endocrine system does not leave when the words stop. It remains in the blood. It remains in the chemistry of sleep that is already claiming you. The melatonin doing what melatonin does. The growth hormone building toward its pulse. The cortisol at the floor of its daily rhythm. You are not just someone who has heard about hormones tonight. You are the hormones. You are the chemistry. You are the body keeping its own faithful, tireless, mostly invisible conversation with itself about how to stay alive, how to stay well, how to move through time with the resources it has. And right now, in this dark, that conversation is finding its night register, slower, softer, turned inward, tending to the things that only darkness and rest allow. Let the chemistry do what it knows how to do. It has always known. Long before you arrived here tonight, it was already at work. And long after the words stop, it will continue.